so far. Number one, we looked at the elimination of CO2, and we do that through the lungs. We looked at the elimination of fecal matter for the purposes of eliminating things that flow through the digestive canals, or through the alimentary canal, and any things that we don't absorb get excreted out. But we do have to look at another mechanism that's tying in the cardiovascular system with excreting wastes, and that is going to be the urinary system. So not only is it going to be excreting waste, this is our way of maintaining water balance. This is our way of regulating blood pressure. The urinary system works really well specifically for that. We'll be able to look at that in this last and final unit. So what I plan to cover today, I plan to cover just the basics to take a look at, again, giving you kind of an anatomical perspective of the kidneys. We'll look into the cross-section of the kidneys and show you where specifically the functional unit takes place. We'll dive into the functional unit, understand the mechanisms behind the nephron, which is our functional unit, point out its role, and then as we go through the nephron, we'll start to understand how we can create urine, we can reabsorb stuff from the urine, and then secrete out any additional wastes. And I'll break each of those down for you. Aim today is to really just cover the anatomical portion and talk about the first function in the nephron. So altogether, what is the role of the urinary system? And the urinary system is basically all for maintaining homeostasis. Not only are we going to be looking at water levels that will regulate our blood volume and blood pressure, but we'll also be able to regulate our pH through the elimination of acids, namely our hydrogen ions. And then also being able to regulate the tonicity of our blood, mainly through adjustment of the osmolarity of the blood. And if you remember, the osmolarity is tied into the amount of solutes in a solution. So we're looking specifically at the stuff that's dissolved in the blood. And what stuff do we have in terms of electrolytes would be sodium, we have our potassium, we've got calcium chloride, we've got phosphate in there, magnesium, and bicarbonate. All these things we need to be able to maintain normal homeostatic values. And again, ever looking at blood labs, you saw the reference values for each of the components in our blood labs. There are also urinalysis that's also done as part of a panel to determine the level of the ions in our urinary system. We'll take a look at how the kidney also reabsorbs glucose. And why are we reabsorbing glucose? We're pretty much dumping fluid in our functional unit, our nephron, and then we're going to have to reabsorb some of that back. And then at the same time, eliminating excess wastes that are also present in our blood. And then we'll also look from a hormonal perspective, the other hormones like renin and erythropoietin, EPO. We don't really talk about the last one, EPO, that much, but nonetheless, we'll begin looking at the renin, angiotensin, aldosterone system. So looking at the kidney, here you can see the kidney. We've taken a frontal slice, and what's prevalent in this particular slice is that you can see two distinct layers. You'll see the renal cortex, which is the outer layer, and then you'll see the renal medulla, which is the inner layer. Now, what you're going to be able to see, if you pay a little attention over to the cortical layer, is you're going to see this functional unit, the nephron, and you can see where that's nestled in. So on the cortical layer, you'll be able to see the formation of our dilute urine. And we'll go into specifically the function of that. We're going to be looking at how we create this dilute urine through the blood vessels surrounding as well as the nephron. You'll understand the system behind that. We'll cover that today. And then when we look at the medullary function, when we move down into the medulla, you'll see how we're going to be able to take that dilute urine and make it much more concentrated. And we do that because we're reabsorbing some of the stuff as it makes its way down. So where you're going to see the formation of the urine, you're going to see it's going to travel down this yellow tube, this collecting tubule, and making its way down to this renal pelvis here, which is the center. And this renal pelvis is the intersection where you're going to see blood flowing in, blood flowing back out, and the collection of this urine that will make its way over to the bladder, down to the urethra, and out into the external environment. So within the renal medulla, you'll be able to see different functional areas. And you'll see different pyramids. These are these pyramids that are in the shape of cones. And these are going to contain the apparatus that are involved in the formation of urine. And then in between these pyramids, you'll see these columns. And these columns are going to contain this fibrous material that's going to anchor the cortex with that of the renal pelvis. And then you're also going to see the flowing of blood, both arteries and veins, through these sections. So really, the functional area that's going to be involved are these pyramids. You have these different, and it varies from person to person, but about 8 to about 18 of these subdivisions that are present. As the urine comes in and you get this concentrated urine, this is going to flow down into these ureters, and these ureters are going to collect into the bladder. And the bladder is going to be able to store that concentrated urine. And so what we're going to be able to see within the bladder is that the bladder will start to fill up. As the kidneys start coming in, dumping in this concentrated urine, you'll see that this urine will start to fill up. And we do have transitional epithelium, which we discussed in our tissue lecture. And transitional epithelium consists of both stratified squamous layers, where some will be squamous, some will be cuboidal, and you get multiple layers where it accommodates the stretchiness of that particular organ. So as these fill up, there's going to be, the next section which we'll be able to see, is as this starts to fill up, this this bladder can hold quite a bit of fluid. So upwards about the 700 milliliters to about a liter of urine. And how we eliminate this urine is through a process called urination, the technical term being micturition. So we notice that there are stretch receptors that are present in the bladder, in the muscle layer of the bladder itself, and specifically more so in the sphincter located down here. So as the pressure starts to increase with the bladder filling up, there are stretch receptors that send signals over to the pons, specifically to this area. It's the spinal area called the pons, where there's a micturition center that receives input involving how much stretch there's taking place in the bladder. And as that fills up and that applies, as that pressure comes in, or the stretch receptors pick up information that's sending over to the pons, the micturition center then sends information back, letting us know to release that urine. So this entire reflex right here, where we get the initial stretch, comes back, sends information back, and causes a release of urine. And this is actually exhibited under voluntary control. We'll talk about this towards the end of this lecture, where the voluntary release of micturition or urination is how we empty the bladder. That emptying of the bladder will result in less stretching that takes place, causing a negative feedback loop. Looking at the urethra, everything from the bladder out to the urethra is how we eliminate our urine. In males, it's also the way that we eliminate semen as well. Now, let's zoom back into the kidney. Again, we'll reserve the process of the formation of urine towards the very end of this lecture. I want to go back and focus back specifically on the kidneys itself. And first thing we need to look at in our kidneys is to look at the blood flow and how we get blood into the kidney and then how we pull blood back out of the kidney through the renal vein. So renal artery is where we start. The renal vein is where blood comes right back out. And then looking at kidney transplants, really the main three areas that you would see for a kidney transplant, really the only connections that need to be made are going to be your renal artery, your renal vein, and then the ureter. And so technically, You can see that we can still live normal lives with one kidney. So if you're ever to donate a kidney to someone who needs it because you need a match, you can still live fairly normal lives. In fact, I've been told, and I haven't been able to verify this through any journals, that really we only need 1/3 of a kidney for at least normal functioning. Okay? But this is going to play a major role when we start to increase cardiac output. That can affect the level of kidney functioning. So as long as assuming that everything is fairly normal, we have a lot of redundancy in how much we need. And in fact, if you were to ever donate a kidney, or if you ever lose a kidney for any reason, what would happen to the other kidney? It would enlarge, and it would assume additional function. So the purpose of the blood flowing through obviously is going to be for the process of filtration. How much of it is going to flow through? You'll be able to see that most of the cardiac output, we're going to get a lot of filtration that will take place in the kidneys. We'll see how it takes place in the renal artery, which is going to be a branch off the descending aorta. And down the descending aorta, the renal artery will come in and supply blood to the kidney for the process of filtration, and then blood will return back through the renal vein. So in that process, let's start from the renal artery and end up at the renal vein. And we know that blood flows through these renal columns. So let's see how we get from the renal artery to the renal columns into the functional unit and back to the vein. And that'll help you understand the blood flow. The second component we need to look at later on is what happens to the fluid that's pushed out of the blood into our functional unit, and how we claim that a good chunk of that fluid back. So we start from the renal arteries. The renal artery is going to come in and branch apart, which we're going to see the segmental artery. And from the segmental artery, we're going to move in between the lobes, in between each of these little conical pyramids. And these are going to be called the interlobar arteries. Okay? So in between the lobes. The interlobar arteries will then branch out. They'll curve around, and they'll become the arcuate arteries. So if they curve around the base of the pyramid, it'll be called the arcuate artery. From the arcuate artery, we're going to branch off into what's called the interlobular artery. Not the interlobar artery, but the interlobular artery. And those are going to be branches off the arcuate artery. From the interlobular arteries, we're going to see now, namely, our functional unit. In this area, from the afferent arterioles over to the peritubular arteries, are going to be the blood supply for the nephron. And we're going to move from the afferent arteriole. We're now going from our arteries to our arterioles. The afferent arteriole will turn into our glomerular capillaries. And from our glomerular capillaries, we're going to branch off again into the efferent arterioles. So we're going from arterial back to capillary to arterial. The efferent arterioles will then move down. You'll see the peritubular capillaries, and I'll show you these when we zoom in. Peritubular capillaries will converge back into the interlobular veins, and we're going back the reverse down the renal column. We're going to go from interlobular to arcuate to interlobar veins, back to the renal vein, in which we're going to get filtered blood back into our systemic circuit. Again, we'll review these arterioles, capillaries, and arterioles. We'll zoom on this when we look at our nephron. Let's take a look at the other functions of the kidney, or our main functions of the kidney. As I mentioned earlier, the excretion of metabolic wastes. The waste that we'll be seeing are things after the metabolism of specific monomers. And so looking at the things that we were able to break down, we were able to break down our main our organic molecules. We've turned our proteins, we've broken down our proteins down to amino acids. We've taken our DNA and broken them down to our specific nucleotides. We had our starches, broke them down to glucose. And I'm missing fats. We've broken our fats into fatty acids. So here we're going to be able to see metabolic waste. When we break down amino acids, one of the byproducts of breaking down amino acids is urea. And urea is produced by ammonia, which is one of our main byproducts from breaking down amino acids. So if you remember our amino acids, we had NH2, C, and then this was a carboxylic acid. And then from here, we had our molecule, and then on top, we had our R group for that particular amino acid. And in the consumption of amino acids, when we break it down, we're going to be able to release this amine group, and that amine group can then join hydrogen ion and create NH3+. And that is ammonia. And ammonia combined with carbon dioxide creates a byproduct called urea. And you'll be able to see how we eliminate urea in the kidney. Creatinine is the breakdown of creatine phosphate. And we haven't talked about creatine phosphate because we didn't really get into muscle physiology. But one of the ways that we were able to store in the muscle, we're able to store ATP is through creatine phosphate. So this is our storage for creatine phosphate. And whenever we break that down, we break apart creatine phosphate, we create something called creatinine. So for any of those that do use any supplements, does anybody use creatine monohydrate here? Yeah. Creatine is exactly what that product is for. It's to increase your storage for ATP in the muscle. Out of all the supplements out there, that was probably the most effective. And if you look at all journal articles that examine most of the supplements out there, caffeine and creatine are typically the most useful, best bang for your buck. And I'm not really trying to... What's that?
And they're the cheapest.
They are the cheapest. And I'm not trying to advertise it. I'm not saying that you should use them. I'm just saying, in general, one of the things that's a big thing about people that take in creatine phosphate, and really just applies for everybody as well, even though we have normal levels of creatine phosphate, the breakdown of creatine, we break it down into a product called creatinine. And creatinine is something that the kidneys need to get rid of. So typically, creatinine is something that we can measure in the urine to determine kidney function. How fast do we break down, given the amount of normal levels of creatine phosphate, how fast can we get rid of creatinine? And there's certain oncology protocols, certain treatment protocols that will look at kidney function, and they base off that how much creatinine is present in the blood. Okay? Present in the blood and present in the urine. The last of these is going to be uric acid. And uric acid is something that is broken down with the breakdown of nucleotides. So we break our DNA down to nucleotides, and once we break down those nucleotides, we have uric acid. And for the most part, it is insoluble in the blood. It is concentrated, and it concentrates mostly in the joints. Does anybody here have gout or know of anybody with gout? Gout is basically an accumulation of uric acids. Our inability to eliminate the accumulation of uric acid results in a condition called gout. And so people that have gout, if you ever hear of anybody, especially in their 30s, maybe late 20s and 30s, which I know a bulk of you are probably entering that age, if you ever start having pain in your right toe, like right at the main toe, more often than not, it's probably either it's an acute injury if you know you injured yourself, but if you didn't injure yourself and you start to get that pain, that's the start of gout. And especially you'll know it after a bout of drinking and red meat. And basically what they'll tell you is to eliminate these foods because you're starting to show symptoms of gout. I know it because I was told that I need to start avoiding it. But hell no, I'm not avoiding it. He gave me a whole list. I remember the doctor telling me, so gout's not that bad. You just got to eliminate some foods out of your diet. And he starts looking through his list. He's like, yeah, you have to get rid of beer, you have to get rid of red meat, you have to get rid of cheese. And I'm looking, I'm like, are you serious? And he's just like, yeah, no, that sucks. I'm sorry.
Just end it there.
What's that?
Just end me.
Yeah, no.
Just go home with some allopurinol and some cultures.
I'm fine. I'm good enough. And if I ever have any, there's prophylactics you can take, and you're not taking away my red meat and my beer. That's going to be a decreased quality of life for me. The other portion of the kidneys, this is our way of eliminating wastes through our urine. The other portion is to also look at our water and salt balance. And all this is going to be related to normalizing blood pressure and blood volumes. Are we going to be able to get rid of excess water if we have it? We're going to be able to get rid of excess salts if it's present. So you'll be able to see this process as we look at the nephron. And you'll be able to see the nephron. You'll see these tubules, and then this descending tubule down over here, which we'll point out later. This whole process is the formation of a filtrate. And again, we're going to go over this today, and how we're going to be able to exchange with the tubules that are wrapping around. The tubules are going to be the capillaries that are going to come in and collect the stuff from the urine that was formed. It's going to collect it and either collect it or not collect it. And this is our way of getting rid of excess stuff. The stuff that's coming out, this portion back right here, is what we're doing when we're making our way back towards the ureters. This is our way of eliminating waste. So this reabsorption process, if we've got too much of something, we'll be able to eliminate it. If we've got too little of it, we'll be able to conserve it. So if you ever have a very salty diet, let's say you have just eaten a really salty broth and you've taken a lot of it out, you notice that after an hour or two, whatever you're eliminating, you may notice how salty your urine is. I don't know how you can really figure that out, but you can at least, I don't know, maybe smell it. I don't know what you would do, but you can tell the composition's a little bit different. This is our way of eliminating excess. You know that if your blood pressure is low, if your blood pressure is low, we have mechanisms in place to be able to conserve that water. And ways we can conserve water is to increase thirst. You can also eliminate sweating. And then the other portion is to use antidiuretic hormone. Antidiuretic hormone will help you conserve water. Okay. So essentially what we're going to be doing is we're going to be reclaiming salts, and then we'll be using antidiuretic hormone to allow us to reabsorb water. So we're going to be involving the reabsorption of water and salts, or the elimination of those two, in order to maintain homeostatic levels of our salts as well as our water volume. Acid-base balance, this is our way of maintaining normal balances of hydrogen ions, and namely our bicarbonate ions. So by now you know your bicarbonate ions are used as a buffer to keep our pH between 7.35 to 7.45. I know all of you guys know that because 100% of you guys got this right on the test. So the whole purpose of this is our way of reabsorbing bicarbonate ions to assist in the buffering capacity of our blood. Anytime we end up with excess hydrogen ions, either through metabolic acidosis or anything like that, we're able to eliminate excess acids through the urine. And there's the mechanisms which we won't really get too much involved, but our way of our ammonia, we combine that with hydrogen ions, we can eliminate those, but at the same time eliminate excess hydrogen ions through that process. And then the secretion of hormones. We still have to look at the hormonal functions, where we're going to see renin as one of the enzymes in the kidneys, which is going to be playing the initial role in the RAS system, our renin-angiotensin-aldosterone system. Okay. And through this process, we would see our ability to maintain salts as well as volume of H2O. Looking at our water balance, I do want to point out, again, looking at the cell. And in the cell, we're going to have our intracellular fluid, and then the water on the outside is going to be our extracellular fluid. And as a recap, we know that the bulk of our extracellular fluid is going to consist of blood flowing through the capillaries, so these are going to make our capillaries, as well as the interstitial fluid surrounding. And within the intracellular fluid, we know the ions that are present are going to be potassium. On the outside, we're going to have glucose, sodium. We'll have also chloride. We'll also have calcium. We also have phosphate and magnesium involved in the inside, which is not really something that we talked about too much. We've got tons of bicarbonate on the outside. So given all of these, we need to be able to maintain normal values of potassium, phosphate, and magnesium within the cell. And so in order to maintain those, we're going to have to exchange with the external environment. And that exchange of the external environment is essentially going to make its way back into the kidneys to eliminate any excess of. So I know one of the quiz questions that I asked you prior was what happens with excess potassium ions. Excess potassium ions that we're not able to eliminate through the urinary system can have a profound effect in terms of the membrane potential of the cell. It can affect the firing of the cell itself. So in those cases, we want to make sure that salt levels are normal within both the intracellular as well as the extracellular fluid. The other portion that we'll be looking at is, again, another recap is to look at the movement of fluid. So we're going to once again take the capillary bed, which I'll draw as a straight line. We have our arterial side, and we have our venous side of the capillary. And we knew through this process, if this were the capillary, we knew that we had a couple pressures in play. We had our blood hydrostatic pressure. Blood hydrostatic pressure is the pressure of the blood within the capillary exerting pressure out. So it wants to push against the endothelium and push out. So this is hydrostatic pressure. Now the other pressure that was involved was blood colloid osmotic pressure. And blood colloid osmotic pressure was the tendency of water wanting to move from low concentration over to a high concentration. So technically what's happening with blood colloid osmotic pressure is the presence of proteins in red blood cells that wants to draw water from a hypotonic solution over to a hypertonic solution. So to draw this out in terms of our U-tube example, here's our semi-permeable membrane right in the middle. We have our solutes that are present on the left side, and we had our two solutes available on the right side. Osmosis is the driving of water from our hypotonic solution over to our hypertonic solution. So we know that the hydrostatic pressure is over to the right. Osmotic pressure, this would be our BCOP, this would be our BHP. The osmotic pressure is the pressure of water movement from the hypotonic side over to the hypertonic side. Blood hydrostatic pressure is due to the buildup of all that fluid on the left side. Now you have this hydrostatic column. This is going to exert a pressure of pushing water back the opposite way. Now, accounting for all these pressures, we know that the movement of water, bulk flow, that takes place on the arterial side, due to the higher pressures of blood hydrostatic pressure compared to that of blood colloid osmotic pressure, we're going to get more net pressure, net filtration pressure, taking place on the arterial side, hence filtration. And due to higher colloid osmotic pressure compared to that of hydrostatic pressure, we're going to get reabsorption. So these pressures that are involved that will result in filtration and reabsorption, the direction of fluid movement we're going to be able to see in the kidneys. So given that, so now we've talked about the movement of water, and then as well as all the stuff that might be dissolved in the water, let's consider those aspects for water input as well as water output. Water intake is driven by the thirst center. So thirst center in the hypothalamus, we do have osmoreceptors that are present that will give us the pressures within that fluid. And the osmotic pressures that are exerted within the fluid that flows through will tell us, and then osmotic pressure again is due to the solute concentration, will drive our thirst. So the saltier the water content in our blood, the increase in the osmoreceptors firing back over to the hypothalamus. The hypothalamus will then, specifically the thirst center, will drive then the need to drink water. So this is going to increase our water intake. So as we start to increase our water intake, that's going to cause distension of the stomach. And the stretch receptors in the stomach will then feed back into the thirst center, letting us know that we've drank enough water. So think about that in terms of how this can be confused for hunger. Because you probably have heard at some point, if you feel hungry again, it could potentially be because you're thirsty. And for those of you that might eat something and still continually feel hungry, well, what's technically happening is that thirst is driven due to the high solute concentration in the blood, which will drive the thirst center. And the thirst center will let you know that you do need to take in water. But take in food, and that will also cause stomach distension. That stomach distension will then inhibit the thirst center, but at least temporarily. But that has not resolved the osmoreceptor still firing back, letting you know you're thirsty. You're temporarily inhibiting that process through stomach distension. So try that out at some point. If you notice that even though you've eaten, you still have that feeling of hunger, it might really just be your thirst intake. The output, so we're taking in water through the alimentary canal. The output, mechanisms for water loss, 60% of that is going to take place through the urine. Defecation, a very small amount of water through defecation. And then the bigger chunk, or second to biggest chunk, would be through the skin and lungs. We eliminate water through the skin through sweating, and lung through water vapor that's lost in, through exhalation. And our way of being able to regulate all those, our output, is specifically looking at the use of antidiuretic hormone. We'll look at ADH, and to a certain extent we'll look also at angiotensin II as well as aldosterone in maintaining normal osmotic pressures. Again, these are going to be related to the salts as well as the volume of H2O. So with that, we've now talked about specifically the functions. We talked about the blood flow. Now we need to take a look at the nephron. So with the nephron, you'll be able to see this is the functional unit. And again, you're going to be able to see the divisions of the kidney. We have our cortex as well as the medulla. So in the cortical layer, you're going to be able to see different parts of the nephron. And we start off with the glomerulus. You're going to see these tubules, and those extend out, travel down into the medulla, come right back up to the cortex, and travel again right back down into the medulla for the creation of concentrated urine. We do have a set number of nephrons in the kidneys, and that doesn't change. The blood vessels that we'll be able to look at, we'll be able to look at the afferent arteriole, we'll look at the capillaries, and then we'll look at the efferent arteriole, and then all the capillaries that come in and wrap around the tubules. If we were to straighten all this out, you'll pretty much, just to simplify, you'll see the nephron, where you're going to see two different areas of the nephron. The renal corpuscle, which is going to be the head of the nephron, which you can see in this process right here. This would be our renal corpuscle. And if I were to straighten out this tube and make it one straight tube, these would be considered our renal tubule. And this is everything down from this point on. So the renal corpuscle, will be able to look at the main components of the corpuscle. We have our glomerular capillaries, which are going to contain the blood. And the glomerular capsule, which will contain our filtrate, which is going to be our dilute urine. It's what we first start off with. So we have to understand the interface between these two for the process of filtration, where we're going to have fluid escape, the glomerular capillaries, and all the stuff that was filtered out of the capillaries are present as filtrate. That filtrate is going to move down the tubule, and the tubule representing the longest portion. This tubule, what we're going to be able to see is that we're going to see what's called reabsorption. Reabsorption is what we're taking out of that dilute urine. There's a bunch of stuff in that urine that we need to recollect. namely water, some ions, as well as the glucose that's present. And we'll talk about what's actually present in that filtrate on the next couple slides. We're reabsorbing stuff that we need, and then anything that we need to get rid of, this is going to be called secretion. This is stuff that's going back from the blood back into the urine. And reabsorption as well as secretion all take place in different areas, and you'll see where those areas specifically take place. So let's zoom in specifically into the renal corpuscle, again, looking at the glomerulus as well as the capillaries present within. The renal corpuscle is composed of our capillaries. The capillaries, again, are between the afferent arteriole. Here's our capillaries that are all bunched up together, and then the efferent arteriole. So we're looking specifically at the capillaries, and then the capsule that's wrapped around. So in that capsule, you're going to see what's also referred to as the Bowman's capsule. The glomerular capsule is going to be a double-walled epithelial capsule that comes in and surrounds these capillaries. And I'll talk about where those double walls exist. You'll see the formation of filtrate as it comes out and forms out and collects in the glomerular capsule and then makes its way down the rest of the tubule. And the first stop will be the proximal convoluted tubule where reabsorption takes place. We're going to have efferent arteriole. Here's our efferent arteriole. That's going to come back in, and that's going to start wrapping around the rest of those tubules, coming in, collecting all the stuff that's present in our initial filtrate. Looking at the glomerulus, here you can see another picture of it. You can see the afferent arteriole. This is blood entering. You'll see the capillaries that are present within, and you'll see there's this whole formation of stuff wrapped around, and I'll talk about that in just a bit. And then you'll see the efferent arteriole. One thing I want to bring up, you're also going to see something I want to point out later, is the distal convoluted tubule. And let me go back to this earlier slide right here, and let me just erase this bit right here. One thing that you'll notice about the tubule, here's our glomerulus. We have the proximal convoluted tubule, and it's called the proximal convoluted tubule because it's convoluted, it's curvy, and it's the closest to the glomerulus. After we leave the proximal convoluted tubule, we'll have what's called the loop of Henle, which dips down. This is going to be the straight trombone-looking like portion of the unit that will dip into the medulla. It'll come right back up. So this whole thing is called the loop of Henle. It loops back into the medulla. We have the descending portion of the loop of Henle, and we have the ascending portion of the loop of Henle. Once we come back, you're going to see the distal convoluted tubule. Again, convoluted because it's twisty, and distal because it's the furthest away from the glomerulus. That distal convoluted tubule comes right back up right next to that glomerulus, which you can see back in this later slide. Distal convoluted tubule comes back up. This area, there's a specific area that we'll reference later called the macula densa, which are going to contain chemoreceptors that will give us feedback about the stuff that's present down the line from the proximal convoluted tubule, down the loop of Henle, back up to the distal convoluted tubule. This will provide good feedback into the glomerulus to let us know how well this whole nephron is working. So pay attention to that when we get to that section. Distal convoluted tubule comes right back up, and then we've got the collecting duct, which will help extract more water to make it down back as concentrated urine back into the renal pelvis. So this area, the capillary, is going to be where filtration occurs. As I mentioned earlier, filtrate is going to be pushed out of the capillaries and will flow down the rest of the tubule. This is again just talking about the afferent arteriole to the capillaries, back out to the efferent arteriole. From this point on, past the efferent arteriole, we're going to have what are called peritubular capillaries. When you get to the book, you're going to see a section of the peritubular capillaries called the vasa recta. Let's see if I have that slide somewhere. The efferent arteriole. Here you're going to see these are going to call the peritubular capillaries. And the capillaries that wrap around the loop of Henle are commonly referred to as the vasa recta. So if you ever see those terms, it might be a little confusing at first. It's just the vessels surrounding the peritubular capillaries. And again, the importance of the vasa recta as well as the peritubular capillaries is to reabsorb the stuff in the filtrate that we need to keep. So going back, let's now focus specifically on the renal corpuscle. And looking at the capillaries, as we mentioned earlier, let's also look at the capsule surrounding, the Bowman's capsule. So in this glomerular capsule, we do have a double layer. And that first layer is going to be the parietal layer, which is the outer layer. This outer layer is present that wraps the outside of the glomerular capillaries. The visceral layer, you can't necessarily see it on here, but the visceral layer is flush right against the glomerular capillaries. And the whole purpose of this visceral layer is to provide a covering for those capillaries. So if you take a look at the visceral layer, notice these dots right here? These dots are called podocytes. They're cells that are part of the internal layer, that visceral layer. And these podocytes have these extensions of the cell membrane. And they literally will wrap around. And you might not be able to see it on this slide because of the color scheme, but you'll definitely see it on this slide over. Here you can see our podocytes, and you can see the extensions of the cells. And you notice how these cells create these slit-like layers? These slits right here are the grates that come in and serve as a protection on the outer layer of the capillaries. You've got fluid moving out of the capillaries. What type of filtration takes place? We have multiple layers that assist in keeping bigger objects within the capillaries. Things that need to stay in the blood are formed elements. You want to keep your red blood cells in there. Things that are dissolved in the plasma. What's dissolved in the plasma? You've got some large proteins. You've got water-soluble hormones. You've got glucose. You've got ions. And you've got tons of water. So looking at the structure of the capillaries as well as the podocytes on the outside, those podocytes, as I mentioned earlier, have these extensions, these finger-like extensions called pedicels. Pedicels are these fingers that come in that literally will lock together like this. And the slits in between my fingers are exactly these filtration slits that you see. So the capillaries are technically fenestrated capillaries. They contain a bunch of pores that allow things that are dissolved in the blood plasma to flow through. Namely, there are proteins, large and small, all the glucose, ions, and water. You also have this basal lamina that's just going to be on the outside of the endothelium that will prevent larger proteins from flowing through. You're going to have these slits that are going to prevent medium proteins from flowing through. And again, the grates right here, the fenestration, the pores, will maintain all the red blood cells and all the other cells contained within the blood inside. So this really helps filter out only plasma with small proteins, really small proteins, but namely things that are dissolved within, our glucose, our ions, and water. And again, this is stuff that is filtered out. So you get the bulk of water, glucose, and ions making its way out. This stuff, most of this we want to reclaim back, and we're going to be able to reabsorb that in the process called reabsorption. So these are the contents in filtration. Some of the other things that we can note in an abnormal function of the kidneys, if you note that any dysfunction of these pedicels, or let's say any mutation of the proteins involved in creation of these pedicels, or any malformation of the fenestrations on here, or the basal lamina, can cause openings that are bigger, and that'll allow larger proteins, that could allow the leakage of red blood cells, and that causes dysfunction of the kidney itself. Now as that filtrate moves out, it's going to enter the space within, between the visceral and the parietal layer, And that filtration that comes in, as I mentioned earlier, will flow back down the tubule. So we're going to have to take a look at the pressures involved. Blood hydrostatic pressure, colloid osmotic pressure. The other new one that we need to look at is capsular hydrostatic pressure. And I'll talk about that in just a bit. So the process for filtration to take place, like you learned earlier, are the pressures that allow filtrate to be produced. This is going to be net fluid pressure out of the capillaries that will push fluid down into the proximal convoluted tubule. As that fluid flows through, I mentioned the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule. Not all nephrons have the same length of the loop of Henle. There are some that extend further down into the medulla, and there are some that don't even really reach the medulla. So, various lengths of the loops of Henle. And typically, general rule is, the longer the loop of Henle, or the longer the pathway of the tubule, the more concentrated the urine becomes. Some additional structures that I do want to talk about that are also going to play a major role are juxtaglomerular cells. And the juxtaglomerular cells are present in the afferent arteriole, that's blood flowing into our glomerular capillaries. Juxtaglomerular cells, are otherwise known as JG cells, are part of this apparatus that detects the blood pressure, particularly within the afferent arteriole. Because what we need to do is we need to maintain normal pressures within the glomerular capsule. Because if we can go back to an early example of what I looked at when we pinpointed the capillaries, I pointed out what would happen if hydrostatic pressure was low. And in the cases where hydrostatic pressure was low, if you remember in our regular capillaries, we had a blood hydrostatic pressure of 35, 25, and then 16. And then our blood colloid osmotic pressure had a pressure of about 25 millimeters of mercury. That was normal pressures. What would happen if blood hydrostatic pressure decreased? Let's bring it down to 25 millimeters of mercury, 15 millimeters of mercury, and then 6 millimeters of mercury. What we ended up with, no filtration. Most of what we saw was reabsorption. So no filtration. And now take that back to our kidneys. If I don't get normalized blood pressure within the glomerular capillaries, the process of filtration will not take place. And if I'm not filtering in the glomerular capillaries, the kidney isn't doing its function with what it's supposed to be doing. It's to create dilute urine and reabsorbing what we need. So that would be, in essence, a form of kidney failure. So in this process, we're going to be able to maintain, and with these additional structures, this is our way of maintaining normal blood pressure and allowing normal filtration to take place. So the JG cells are present within. There's these cells contained within the JG cells that detect pressure. So there's stretch receptors that are located within the smooth muscle surrounding the afferent arteriole. And I'll talk about that at the end of today's lecture. Mesangial cells are present in between the capillaries of the glomerular capillaries and right before the capillaries. There's two different types of mesangial cells, but for the most part, they work the same way. Mesangial cells, think of them as a net. You've got the afferent arteriole, and then you've got all these capillaries that are present within the glomerular cup or within the renal corpuscle. In order to decrease the surface area to slow down the filtration process, I can retract some of those capillaries. If I want to increase filtration, I can open up those capillaries up even further. So these mesangial cells help contract and relax to either pull back the capillaries or allow them to fully expand. The other portion is called the macula densa. And the macula densa are chemoreceptor-filled cells that are part of the distal convoluted tubule. And they come on and they provide feedback that to the juxtaglomerular apparatus that will help regulate flow of blood into this area. And what the macula densa specifically detects is the salt concentrations. They're looking specifically at sodium as well as chloride ions. So this is our indirect, which I'll talk about later. This is our indirect measure. This is our direct measure of blood pressure. This is our indirect blood pressure and blood flow. So tying this all together, we have, once again, our nephron, which is going to consist of our renal corpuscle as well as our renal tubule. And then also the blood supply that we mentioned earlier in the very first beginning. Everything from our renal artery feeding into the afferent arterial, glomerular capillaries, efferent arterial, and the peritubular capillaries surrounding the rest of the tubule. That comes in and collects right back into our veins to feed back into the returning systemic circuit. So glomerular filtration, as I mentioned earlier, the afferent arterial is going to be feeding blood into the glomerular capillaries. As that blood is flowing from the afferent arterial to the glomerular capillaries, you're going to see the pushing out some of the filtrate out. And that filtrate is going to be stuff that is present in the capillaries that'll make its way into the glomerular capsule, but not all the plasma from the blood is going to be filtered. Meaning that we're going to see blood flowing through, we're going to be able to push some fluid out, but the rest is going to make its way down the efferent arterial. So this is our first portion, again called filtration. And that filtration is the primary process at which the kidneys function to filter the blood out. And how much filtration actually takes place? It's about 150 liters per day in females. It's about 180 liters per day in males. So if you take into account about 5 liters of blood, that comes to about 10 times amount of filtration of that blood. We're filtering your blood at least 10 times per day. So when you think about this filtration that's provided by the kidneys, and take into consideration people that have kidney failure, they typically would have to undergo what's called dialysis. A dialysis machine is something that they need to connect to in order to filter their blood manually. So typically in those cases, they'll be able to tap into an artery and tap into a vein. Blood will flow out the artery, flow into a machine that will manually filter out the blood to feed it back into the vein and remove those components manually. So with that, when we think about how much fluid flows out in filtrate, again, about 180 liters flows out per day. As that fluid makes its way through, we're going to be able to reabsorb that fluid. Imagine peeing out 180 liters per day. That's a lot of fluid. We want to be able to reabsorb about 99% of that. And as we reclaim about 99%, so 99% reabsorption, how much urine output are we going to be able to see a day? About 1 to 2 liters of urine, concentrated urine. So all the stuff that we don't want with that leftover fluid of water is urine. So looking at these, I'm going to talk about the pressures on this slide, but all the information that you need is available on the next slide over. So the 2 pressures that we talked about already, blood hydrostatic pressure and blood colloid osmotic pressure. The pressures are going to be slightly different in here because we're looking at the pressures in the afferent arterial into the glomerular capillaries. And the blood hydrostatic pressure, the pressure in that area, is going to be 55 millimeters of mercury. Again, blood hydrostatic pressure within the arterioles into the capillaries. Blood colloid osmotic pressure, again, is the osmotic pressure. That's the due to the presence of all the proteins and red blood cells, water wants to move from the hypotonic solution on the outside of the capillaries into the cell. And that blood colloid osmotic pressure is 30 millimeters of mercury. It was 25 that we learned about in the systemic capillaries. It's about 30 specifically within the glomerular cup. So 55 and then 30. The other thing that we need to talk about is the capsular hydrostatic pressure. And what exactly is a capsular hydrostatic pressure? You have fluid exiting the capillaries. And it's not just freely moving around, it's actually collected in the visceral layer. Okay? That visceral layer on the outside is going to start to fill up, and it's going to increase the pressure in the capsule. So that pressure, we've got pressure on the inside due to the blood, we've got pressure on the outside due to the formation of filtrate. That pressure on the outside is going to counter that of the glomerular hydrostatic pressure. So if I've got pressure pushing out to push blood out, I've got another pressure forming within the Bowman space that's wanting to push fluid back in. So the glomerular blood hydrostatic pressure, which is 55 millimeters of mercury, is going to be opposed by two pressures. Again, number one would be the capsular hydrostatic pressure, which is about 15 millimeters of mercury, minus 30 millimeters of mercury for the blood colloid osmotic pressure. So 55 minus 15 is 40. 40 minus 30 is 10 millimeters of mercury. And that is the net filtration pressure. That is the pressure, net pressure being exerted out to push filtrate or fluid out of the capillaries into the Bowman space to flow down into the proximal convoluted tubule. What questions do we have on the pressures?
You said for number 3 was hypotonic to...
Yeah, so think about what's present in the filtrate. It's mostly water, we've got glucose, we've got ions in there. In the blood, we've got red blood cells, we've got large proteins and small proteins. Because I've got more stuff dissolved in the blood, water is going to want to move from a hypotonic, this is hypotonic compared to this, which is hypertonic. So water wants to move in that direction. And again, drawing out that YouTube example earlier, I've got more solutes on my left side, and I've got less solutes on the right. The direction of water movement is towards the left. So the left represents the capillary. On the outside represents the Bowman space. That's filled with filtrate. So this pressure right here represents blood colloid osmotic pressure. The pressure out to the right is our GBHB, that's our glomerular blood hydrostatic pressure. The other pressure that's also involved is capsular hydrostatic pressure. And that's due to the pressure pushing back to the left as well. And again, that's the pressure built up of fluid on the outer, wanting to push right back in. Does that help? Our glomerular filtration rate. And each of these are described for you. Blood hydrostatic pressure, capsular hydrostatic pressure, exerted against the filtration membrane by fluid in the capsular space, and then blood colloid osmotic pressure. So those haven't changed. All of these forces, the balance of all these forces, are Starling's forces. The same forces that you learned in the heart, same things about preload, or the filling of the heart due to preload, it's going to cause a bigger contraction. Same thing with these forces. These are all the forces that are involved in the process of filtration. So given that, again, thinking about that amount of pressure, 10 millimeters of mercury for the net filtration pressure. That excess pressure, all those pressures over the course of the day cause this much filtration. So normal filtration for glomerular filtration, you'll see how much this is filtered, we'll be able to show you what the normal values are. Looking at all of this, as you now understand the pressures involved, we know that the pressures involved are going to be fairly regulated. We want to be able to maintain normal pressures. And if our blood hydrostatic pressure is low due to decreased blood pressure or hypotension, we want to be able to regulate normal blood flow through the kidney. Otherwise, if the kidney isn't operating properly, we have malfunction of the kidney, and therefore we're going to have kidney failure at that point. So we need to regulate the GFR, the glomerular filtration rate, in order to maintain normal flow through the glomerulus. So how do we do that? How do we regulate the glomerular filtration rate? There's two mechanisms involved. I can either adjust the blood flow in here into this apparatus, the blood flow through the afferent arteriole and the glomerular capillaries, to the blood flowing out through the efferent arteriole. And there are nozzles that I can use, and they're technically not nozzles, but I can vasodilate and vasoconstrict to increase and decrease blood flow. The same mechanisms that you learned in our vessel lecture in increasing and decreasing blood flow to the tissues, we can do the same thing to the kidneys. The other mechanism that's present, altering the glomerular capillary surface area available for filtration. And what I mean by that is how much of the capillaries are exposed to allow filtration. If I want to reduce the amount of filtration, I can pull them back and reduce the surface area of all these capillaries. If I want to increase filtration, I can open it right back up. So those are the mechanisms. These are the efferent ways of adjusting the glomerular filtration rate. And we'll be able to look at three types of mechanisms. Renal autoregulation is related to the kidneys and how we can regulate the glomerular filtration rate. So this is autoregulation. Why? Because the kidneys are regulating it themselves. This is an intrinsic mechanism. Kidney does it all by itself. The other two are extrinsic, meaning that they're going to involve external factors. Neural regulation is going to involve the autonomic nervous system, where we're going to be able to see parasympathetic and sympathetic pathways that will adjust the blood flow into the glomerular capillaries as well as the glomerular filtration rate. And the hormonal regulation is going to involve the other hormones, namely angiotensin II as well as atrial natriuretic peptide. Angiotensin II was our potent vasoconstrictor. And then ANP was our potent vasodilator. So again, these two have antagonistic effects with one another. We'll be able to see all of these on the next slide over. Renal autoregulation. So we're going to talk about the reflexes involved. This is all feeding information back into the kidneys and the efferent pathway outwards to regulate renal blood flow. And the two mechanisms here are in place. The first one is called the myogenic mechanism. And the other second one is tubuloglomerular feedback. The first one, in a gist, involves blood flow. Right? So this one involves blood flow. And blood flow would be direct in this case. This gives us ion concentration that will be able to infer the amount of blood flow. This is an indirect path. This is basically saying how salty is the urine, and depending on the saltiness will give us an idea of the amount of blood flow through the afferent arteriole. So the myogenic mechanism is entirely a negative feedback mechanism. Now think about what happens when, let's say, you have a hose, right? You've got a hose, you've got a valve that's flowing water through that hose. You don't want too much water flowing through it, because then your hose is flopping all over the place and can cause damage to the hose itself. You don't want too little of it, because now you've got too little of the blood flowing through. You want to have just about the right amount. So technically, the glomerulus wants an optimal level. It doesn't want too high, it doesn't want too low, but can accommodate a good amount of flow through glomerular apparatus. So as you take that hose and you turn it up quite a bit, that pressure is going to stretch out the walls of the hose. And we don't want that to happen. So technically, what's happening in the afferent arteriole is that as blood flow, let me go back to an earlier slide. As you get too much blood flowing through, I've got high blood pressure and therefore high blood flow through, that's going to stretch out the JG cells, or juxtaglomerular cells. They'll stretch out, and there's stretch receptors that will pick out the level of stretch. So in order to reduce the amount of blood flow, that stretching will trigger the contraction of the smooth muscle cells. It'll come in, due to the stretch, will cause the smooth muscle cells to now vasoconstrict. And that vasoconstriction will reduce the amount of blood flow into that area. So what started off with too much blood flowing involves vasoconstriction, which will reduce the amount of blood flow. Does that make sense? The second mechanism is the tubuloglomerular feedback. Again, going back to our earlier slide, and the macula densa here is from the distal convoluted tubule that wraps all the way back up. And let me go back to an earlier slide. Here you can see the distal convoluted tubule coming right back up, right near the glomerular cup. And this section right here has these macula densa. The macula densa have these chemoreceptors that detect the salt concentration. And if the salt concentration is off, it can come in and cause vasodilation. And the vasodilation is provided by nitric oxide. So if I inhibit the amount of nitric oxide, which is a potent vasodilator, the vasodilator, if I inhibit the vasodilator, that's going to cause vasoconstriction. Again, all related to the ion concentration. And the mechanism is fairly complex, so I don't want to get into too much detail about it. But all it's really saying is that if the ion concentration's off, we need to adjust via nitric oxide. So that covers renal autoregulation. Again, regulation that's all done by the kidney all by itself. At no point did I ever say autonomic nervous system involved, there's no other hormones coming into play, the kidney can do it all by itself. Neural regulation is all through the autonomic pathways. And looking at these, the sympathetic pathways release norepinephrine. We already established that the postganglion fibers that come in and terminate, so here's our postganglion sympathetic neurons that terminate on the afferent arteriole. They're going to be releasing norepinephrine. We have adrenergic receptors on these smooth muscle cells. And those adrenergic receptors receive norepinephrine and cause vasoconstriction. If you can go back to an earlier slide, specifically when we talked about cholinergic and adrenergic receptors. Adrenergic receptors are the ones that pick up epinephrine and norepinephrine. Some cells respond by vasodilation. Some cells respond by vasoconstriction. Okay. The adrenergic receptors here for the smooth cells, when they're activated, cause contraction. So the contraction that takes place on the smooth muscle cells surrounding the afferent arteriole causes vasoconstriction. Why is vasoconstriction important? Because vasoconstriction also increases the blood pressure in that area. When we increase blood pressure, that's going to be able to maintain, or actually technically increase the filtration rate. So we want to be able to maintain, and the best case example is to look at what happens when we have decreased blood volume or decreased blood pressure. And a good example of that would be hemorrhage, loss of blood, or loss of blood volume. Any drops in blood pressure, we need to increase blood pressure increase blood pressure in order to maintain normal kidney function. The last of these is to look at hormonal regulation, and the two hormones that are involved, angiotensin II and ANP. Angiotensin II being, again, the potent vasoconstrictor, and what that does is to reduce the glomerular filtration rate. So not only are we constricting the input for the glomerular capillaries, we're also going to constrict the output of the glomerular capillaries. Meaning all I need to do is squeeze both of these sides, and that's going to reduce the amount of blood flow through the glomerular capillaries. In effect, reducing the GFR. Atrial natriuretic peptide, remember again, when blood flows through the atria of the heart, is going to release ANP. So ANP comes out and causes relaxation. Where does ANP take effect? And I did talk about ANP as being a potent vasodilator. It specifically works on the mesangial cells. And again, the mesangial cells are present in between the capillaries. So that's going to increase the surface area. It's going to open up as much of the capillaries as possible to increase the filtration rate. So this raises the GFR. This reduces the GFR. Again, another mechanism as to how these are antagonistic hormones. One was a vasoconstrictor, the one was a vasodilator. Now you know the mechanism of how these two work. They work specifically on the kidney level. So that completes the loop of these hormones, and then we'll talk more about the RAS system in more detail in terms of the tubule. All we've talked about so far, keep in mind, all we've talked about so far is the glomerulus for the purpose of filtration. So now that we have an idea of what's happening in the glomerulus for the purposes of filtration, GFR is one of the values that we're looking at in terms of kidney function. And the normal GFR for men is about 125 milliliters per minute in males. 105 milliliters per minute in females. This is how much filtrate is formed in the glomerular capillaries. We don't want an excess of that, because that's going to cause a lot of filtrate forming through, and they could end up destroying some of that, the pedicels and the sensitive filtration system that we have in place. We don't want too little, because then that's not good kidney filtration that's taking place. So our way of being able to measure normal values, we can estimate based on urinalysis. And typically they would look at creatinine values. They look at serum creatinine, and then creatinine in the urine, and they can estimate the glomerular filtration rate. And anything below this is indicative of kidney disease. And we see different stages at which we see decreased glomerular filtration rate. Let's start with the most extreme version, kidney failure. If you have a very low GFR, that means no filtration, very limited filtration is taking place in the kidneys. If your kidneys can't filter, you'll have to rely on an external dialysis machine to do the filtering for you. And as we start to move in lesser amounts of decrease in the glomerular filtration rate, you can see the different stages. So anything greater than 90, but lower than our normative values, will indicate stage 1 kidney disease. Okay, so you have slight kidney damage with a normal glomerular filtration rate. So typically they won't relate it kidney damage due to the glomerular filtration rate. They're looking at some abnormal urinalysis of some kind, something else that's indicating kidney disease. Any more decreases in the GFR relate to stage 2, 3, 4, and then 5 of kidney disease. And again, that's the dysfunction of the kidney in terms of filtration. End-stage renal failure is basically stage 5. So that point, your kidneys are pretty much on the last legs. They're failing, and you're probably put on dialysis. you would need a kidney transplant at that point. That's pretty much it. So far, all we've talked about is filtration. Again, all this is related to filtration, but going back to, we need to talk about reabsorption, and we need to talk about secretion. Again, all we've talked about is the fluid flowing through the glomerular capsule, out into the tubule. We're going to need to reabsorb that stuff back in. All the good stuff that was present in the filtrate, our glucose, our ions, our water, needs to get reabsorbed back into the blood. All the stuff that we need to get rid of afterwards, that's present in the blood that we didn't get rid of in the first place, is tubular secretion. That's stuff that's going back into the urine, and stuff that we'll talk about on Thursday. The nephron. So I gave you an overview of the anatomical structures, and we went specifically over the functional unit, which we call the nephron. And looking at the nephron, we are going to focus specifically on our vessels, and let me pull up a slide that will help kind of sum that up for us. This was our simplified nephron that you were able to see, and we want to focus specifically on the nephron. Here you can see the renal corpuscle, which is going to have our glomerular or Bowman's capsule. And this is going to be the beginning of filtration, where we're going to have fluid moving from the capillaries, and the capillaries supplied by the afferent arteriole to the glomerular capillaries back to the efferent arteriole. We get filtration that takes place that's going to push fluid out using those pressures that we discussed on Tuesday, namely our glomerular blood hydrostatic pressure minus our capsular hydrostatic pressure minus the blood colloid osmotic pressure. That gives us a net pressure of 10 millimeters of mercury that pushes fluid out. So this is going to be our preliminary urine. This is our filtrate that comes out. And remember, from the structure of the glomerular capillary, specifically the visceral layer of the glomerular capsule, you saw the presence of our podocytes. And these were structures that have these pedicels that reach out and they create these filtration slits. And these filtration slits only allow small molecules out. And so those small molecules that you're going to be able to see are going to be our ions, you'll see water, sugar, but any of the bigger stuff, their cells, our bigger proteins, are going to remain within the circulatory system. Okay? So anything that doesn't get filtered out in the glomerular capillaries makes their way back into the efferent arteriole, which is going to circulate back, is going to wrap around the rest of our tubule, which will start the process of reabsorption. So whatever stuff we've pushed out, remember, we've pushed out a lot of water, we pushed out a lot of glucose, we pushed out a lot of ions. We want to be able to pull that stuff out. Because if you end up peeing this out, we're going to end up losing about 150 liters per day. And that's not what we want. We want to be able to reabsorb the stuff that's absolutely necessary. And how much we reabsorb is going to be dependent on certain things. We're going to be looking at whether the sympathetic nervous system is fired up. We're going to be looking at blood volume. And that's stuff we're going to be examining today. Reabsorption as it takes place down the renal tubule. And the areas that we need to look at for the renal tubule, we'll be specifically looking at the proximal convoluted tubule. We'll look at the loop of Henle, the distal convoluted tubule, and then the collecting duct. And we'll be able to look at reabsorption as it takes place, because as you can expect, it differs from section to section. Not only are we looking at things that are close to the glomerulus, you're going to see the convoluted tubule, you'll see the distal convoluted tubule. You saw also on Tuesday that the distal convoluted tubule comes back and provides feedback to the glomerulus by virtue of the macula densa. The macula densa has chemoreceptors that detects the content of the filtrate by the time it makes its way over to the distal convoluted tubule. Then it can provide some feedback saying, well, it doesn't look like you have enough fluid coming out, so let's do some readjustments here so that by the time that fluid makes its way back, it's going to correct itself. That's all part of the renal autoregulation that we learned about on Tuesday. And then as we go through, so what you can be expecting is reabsorption taking place throughout the entire, we're going to be absorbing a lot of water, reabsorbing a lot of glucose, reabsorbing a lot of the ions, namely our sodium, our chloride, a bunch of stuff that we'll talk about as we make our way down, so that by the time we make it to the end of the collecting duct, down to the papillary duct where the renal pelvis is. Remember, our renal pelvis is where the renal artery came in, the renal vein came in, as well as that concentrated urine. Doctors typically call this the sort of bladder of the kidney. This is where you get the most concentrated urine that comes out that's going to get excreted out or urinated out. So looking at that 150 to 180 liters that gets filtered out, you'll be able to see how we reabsorb all the way through to produce that concentrated urine that you produce on a regular basis. So that should provide a good overview of what we talked about on Tuesday. What we're going to do is going to continue on. We've already talked about filtration. Let's take a look at what happens with reabsorption all the way through. The last thing I didn't quite mention is secretion. So not only are we going to be reabsorbing, but anything that's also present in the blood that wasn't reabsorbed, that wasn't filtered out, may get secreted out at different areas of the tubule itself. So this is the formation, this is the presence of what's called secretion. So in effect, looking at how to sum up urine, as I mentioned on Tuesday, what's actually urinated is what's filtered minus what's reabsorbed in the tubule, reabsorbed, that of what is secreted back into the tubule. And that's going to give us a concentrated urine or maybe even a dilute urine. That really inherently depends on how much blood volume, how much water you've had to drink. Okay, as you know, this is probably fairly obvious to you. If you drink a bunch of water, you may notice you probably have dilute urine. If you haven't had any water, let's say you wake up in the morning, you're fairly dehydrated, and you urinate, you may notice that it's probably dark in color, probably has a very strong odor to it. So you can see the different composition in your urine, and all that depends on, number one, your salt content, but number two, mainly the amount of water that you've had to drink. All right, so with that, we're going to move past our GFR regulation, our glomerular filtration rate. Now we're going to focus back on tubular reabsorption. Okay? So as I mentioned on Tuesday, filtration takes place in the glomerular capillaries, but not all is filtered out. That doesn't mean that all the plasma and all the water and all the ions gets filtered out. You get a good chunk of it that gets sent out, but you're going to now have this thicker blood that makes its way through. Okay? So it's this duty right here. This is the stuff that's going to return back into the renal vein down the line. And so when we go for the peritubular capillaries, we're going to want to reabsorb all that fluid back from our preliminary urine or filtrate and so forth. So we'll talk about this point here. So now that gives you an idea of what's present. You know that we've got red blood cells, we've got large proteins, we've got smaller proteins, we've got hormones, stuff that's present in our efferent arteriole. Now that you know the compositions in our filtrate versus what's present in our efferent arteriole, let's take a look at, once again, our arrangement. You have our proximal convoluted tubule, again, proximal because it's closest to the glomerulus, loop of Henle, which dips out into the medulla, which is more the core of the kidney. We're going to loop right back up. Here we have the descending limb, and then we have the loop, and then we have the ascending limb. As you can expect, we're going to see different characteristics to the structures of these different loops that will lend itself to the different absorption that takes place down in the descending arm versus the ascending arm. We'll curve right back up into the distal convoluted tubule, then back down the collecting duct. Okay. Now that talks about the movement of the filtrate to urine, but we also have to talk about the capillaries. And the capillaries that are present. Here we have our efferent arteriole, and then we're going to wrap around this whole thing. And notice I am kind of drawing a little bit backwards in that area. You're going to see the peritubular capillaries. So we go from the efferent arteriole back to peritubular capillaries where we come in and allow the collection. So you literally have these capillaries that wrap around that assist, well, not necessarily assist, but they are the end point for reabsorption. We're reabsorbing stuff from the filtrate back into the capillaries. And I did draw it a little bit backwards over here. I did go from the proximal convoluted tubule back to the ascending limb, back down this way, back to the descending limb, and back up and around. And you'll see that as I drew the filtrate moving down this way of the loop, you see that blood will flow the other way. And I'll have a term for that that'll explain that later. Now, the difference between the paratubular capillaries and the vasa recta is just really by the nomenclature. Paratubular capillaries represents the capillaries surrounding the entire nephron, namely the PCT and the, I'll call them the PCT and the DCT. The vasa recta is the capillaries surrounding the loop of Henle. Again, just a naming convention. There's a little bit of an anatomical difference, but that's really a moot point. Now, tubular reabsorption. How much do we reabsorb? About 99% of what was present in the glomerular filtrate is going to be reabsorbed. So as I mentioned, about 150 to 180 liters per day. And we're going to reabsorb 99% of that, which is going to bring us to about 1 to 1.5 liters per day in urine. So we're going to have to reabsorb a lot. And what are we reabsorbing? Again, things that were filtered out in the glomerulus. Glucose made its way through, electrolytes, vitamins, water, amino acids, and any other small proteins that made its way out of the glomerulus need to get filtered back into the capillaries. And where does most of the reabsorption take place? Right in the proximal convoluted tubule. It should make sense because this is really the first output. From the glomerular filtrate, we're going to go through the proximal convoluted tubule. How much of it's going to get absorbed by the end of the proximal convoluted tubule? About 65% of it. So almost 2/3 of the stuff. And what stuff am I talking about? Mostly water. Along with these other essentials that I mentioned on the prior slide. Sodium, chloride. We're going to have bicarbonate, glucose, amino acids, and those vitamins are going to get reabsorbed. So about 2/3 of that will be reabsorbed. Notice that on the slide to the right, and this you can reference because I really particularly like this image because it helps you point out different areas. Again, here's our glomerulus. Here you can see the PCT. Here's our DCT. Here is our loop. And then our collecting duct. And this slide talks about things that are going to be reabsorbed in blue and our secretion that's going to take place in green. So this is a really good summary. And at the very end of this section, I also do have another good summary in the form of a table that pinpoints where reabsorption takes place and where secretion takes place. So like I said, 2/3 of the stuff that I mentioned here get reabsorbed in the proximal convoluted tubule. But there is a little bit of secretion that takes place as well. Hydrogen ions, you'll see ammonium, stuff that we get rid of, and then also some drugs here. And I don't really point out any drugs that are secreted at this point, but any things that we do need to get rid of, some are eliminated at this point. The other portions we'll talk about, I'll talk about when we get to the loop of Henle. You'll see the key difference, and again, I'll talk about this later. The descending, you'll see the reabsorption, water. And that's the only thing that's reabsorbed in the descending limb. In the ascending limb, the only thing we're reabsorbing, salts, sodium, potassium, and chloride. So that's going to be the main difference. But the big chunk of where we reabsorb is in the proximal convoluted tubule. The next thing to look at is when we make our way into the DCT. Again, a quick gist of reabsorption. Salts are reabsorbed, sodium, chloride, and calcium in the DCT. And then in the collecting duct, more salts. We're reabsorbing salts and we're reabsorbing water. So that's really the big chunk of it. So if you're looking at where we're reabsorbing most of the stuff that we were able to absorb through digestion, really in the PCT. And then everywhere else, we're fine-tuning and adjusting. And so again, depending on water levels, depending on electrolyte levels, you're going to see that hormones are going to come into play that will alter the composition of the urine through reabsorption, especially towards the latter end, the DCT and the collecting duct. So let's take a look on the next slide over, looking at water reabsorption via the osmotic gradient. And the key thing about this slide, if I could summarize this in a couple words, is that whenever you have salt move out, let's say salt being reabsorbed, water typically follows with it as well. So let me show you a diagram, or at least let's start to draw out a diagram that we've always seen. And I always reference this YouTube because it's very physiologically relevant. And as I mentioned earlier, we have our solutes in our solution, and on the other side, we've got less solutes. Let me just draw 3 on this one, and I'll draw, I guess it really doesn't matter. Let me draw like 6 or 7 of them on the left side. So on our left, we're going to have a hyperosmotic, and on the left, hypoosmotic in relation to one another. So the direction of osmosis, well, you guys answer that. Which direction would you see water moving? Would you see it moving to the left or moving to the right?
To the left.
To the left, right?
Yes.
Why is it moving to the left? Mainly because it's trying to, again, equalize the concentration on both sides. Here you see a lot more solutes on this side. Over here, you see less solutes. So in order to balance out, osmosis wants to move over to the left. Okay, I'm not going to go into hydrostatic pressure because that's not really the focus, even though that does apply. So if I were to, now looking back at the semi-permeable membrane, every time I've talked about the semi-permeable membrane, I've always said that the solutes don't move. But what if I could get those solutes to move? What if I could transport some of these solutes to the other side? And let's just say, due to osmosis, we see the water levels move. We see the water levels rise up to that point, and the water levels, we'll bring the water levels back. No, don't get rid of that. We'll bring the water levels back over here. So now the concentrations are balanced out. We've definitely got more water over here. This whole thing is in now net osmotic balance. We have equal amounts of water moving to the left due to osmotic pressure. We have equal amounts of water molecules moving to the right due to hydrostatic pressure. Now, as I mentioned, this semi-permeable membrane, if I could move solutes over, and I can move them over, if I can move that solute over from the left over to the right, which direction would water move?
To the right.
It'd move over to the right. So anytime I'm going to start pushing those solutes over, now water wants to move back this way. And that's exactly what you're going to be able to see through reabsorption. The general principle, especially in the proximal convoluted tubule, you're going to see coupling of the movement of salts. Anytime you move salt across, water is going to want to move as well. Okay, so that's coupled movement. And that's exactly what we're talking about with this slide, is water reabsorption due to the osmotic gradient. Whenever we move salts or any of the ions across, that's going to allow this, it's going to increase the osmotic gradient as we've been able to do on this image to the top. This whole movement of water is passive. I'm not using ATP to move water across. The osmotic gradient is just increasing the tendency for water to move over from the left to the right as we move solutes over. And now, water will move through osmosis, so it'll diffuse across that semi-permeable membrane, but what's going to enhance the movement of water? How do I get water to move faster through? Again, from our tissue lecture, way back when, if you can recall, I mentioned aquaporins. Aquaporins were water channels. So the presence of aquaporins helps water move faster. Okay? So with that, all I would need to do to get water to move across is to build out some aquaporins. So you're going to be able to see a hormone that's going to come in and activate these aquaporins. It's going to create aquaporins that will increase these channels to shuttle water across. Because it's exactly what we want to do for reabsorption. We want to be able to reabsorb water fairly quickly. We don't want it to just diffuse across and trickle through. We want to move it. Where is that going to take place? Namely in the proximal convoluted tubule as well as the loop of Henle. All areas that we'll talk about in just a bit. So in this image to the right, here you can see the movement of salts, our sodium and our chloride, and then you're going to see water following as a whole. When we talk about the reabsorption of all the stuff that we've just talked about, when we look at sodium chloride bicarbonate, glucose, amino acids, and vitamins, we're going to have to be able to move these solutes through both active and passive means. Again, active. What did active movement require? Active transport required one specific molecule that I quizzed you guys on. What was that?
ATP.
ATP. We have to use ATP to shuttle it across against its concentration gradient. In passive transport basically is just moving down its concentration gradient from a high to a low concentration. We're going to be able to look at the different transporters that we have to move these ions across. So again, in the proximal convoluted tubule, move these ions across and water will follow. So ion transport is what we'll talk about the next couple slides over. Water will just follow along down its osmotic gradient. Again, moving salts over, moving ions over will cause water to move along as well. All right. So which things are we going to be looking at? Again, sodium, chloride, we'll be looking at calcium. To a certain degree, we'll look at hydrogen ions. This is going to be more related to acid-base balance. This is our bicarbonate. That you should be familiar with by now. Phosphate is going to come in also, and I will point that out, although I won't really require you to remember phosphate. You'll also see sulfate come in as well, and ammonia. All right, so let's take a look at how this takes place. So from many slides from this point on, you're going to start seeing these types of images. And to quickly orient you what you're looking at, you're looking here at the lumen of the tubule. You're going to be able to see the epithelial cells, or tubule cells. Here you'll see the interstitial fluid, which I'll name out IF. And that interstitial fluid, all this will consist of the extracellular fluid. And here in the peritubular capillary, obviously this is our capillary. So the direction of reabsorption is always going to move from left to right in these cases. We're trying to reabsorb fluid as well as all the stuff that we've mentioned, all of our salts, sodium, chloride, everything else is going to move from left over to right and reabsorbed into the capillary. So there's only two ways to be able to get stuff across. Either you get it in between the cells, which we call paracellular reabsorption, or we get it through the cell, which we call transcellular reabsorption. The mechanism behind paracellular reabsorption is basically in between. And here you have our tubule cells, which are connected together by tight junctions. It does allow things like sodium to move across. It holds fairly tightly, but sodium can leak across. And typically in this case, we're going to move from a high concentration over down to a low concentration. There's no transporters, there's no leaky channels or anything like that's present at the tight junction. It literally will just flow right in between the cells and leak right through. So this typically is, or in all cases, going to be utilizing passive transport. Again, this is all taking place in the proximal convoluted tubule, but specifically within the first half of the PCT. So we'll be looking specifically at the movement of ions. Now, stuff that moves across the cell is transcellular reabsorption. And the way we have to get sodium across the cell is to shuttle it across. So where we would see the level of sodium, and again, remember where we had in our generic cell when we talked in cellular physiology, where was most of our sodium? Think of the generic cell. Where was most of our sodium? Was it on the outside of the cell or the inside of the cell?
Outside.
Outside of the cell, right? So in order to get that sodium out into the outside, we had to utilize a pump. And what pump did we use?
Sodium pump. Sodium
I heard potassium, I heard sodium. Sodium-potassium pump, right? So we had to use ATP to pump sodium against its gradient from a low concentration over to a high concentration. As we move from a high concentration, now we can shuttle that sodium, which is now high in the interstitial fluid, out into the capillary. So as I start shuttling things across, now sodium, which is high in the lumen, can now just diffuse across. So this is going to be a form of passive. We're going to be able to see that sodium can now diffuse across down its gradient from high to low, and then from low to high. So all this employs both passive and active means to get things across. And again, all this slide talks about is just sodium. But the key thing with sodium, whenever we move sodium, there's going to be a bunch of symporters and antiporters. These are typically called co-transporters. Co-transporters means if I'm allowing sodium in, well, in the case of a symporter, if I'm going to allow sodium in, I'm also going to allow something in as well. It takes two to basically move through from the extracellular fluid, in this case the lumen, out into the cell. An antiporter means sodium in and something else moving out. So these are the different gates that we're going to have to get things across. And again, all this taking place in the proximal convoluted tubule. The symporter in this case would require movement of sodium at the same time the movement of glucose. So this is our way of getting sodium in and glucose in. And glucose will then move down its concentration gradient across this transporter, and it'll go from the glucose will go from inside the cell into the interstitial fluid, into the capillary. Again, we'll see our sodium-potassium pump, where we got sodium into the cell. Now we got to pump sodium back out into a high concentration, back into the capillary. So this is an example of two things moving together, sodium and glucose. The antiporter, you're going to be able to see on the next slide over. And in this case, let me just quickly describe what's happening. Sodium going in, hydrogen going out. And that should make sense, because think about what we're doing. Again, reabsorption moving over to the right. We want to reabsorb, we want to conserve sodium, but we want to get rid of excess hydrogen ions. So this is a form of secretion. We're dumping stuff into the lumen. But we're not just dumping hydrogen ions. Hydrogen ions don't just sit around in the blood. Right? They don't just sit there freely float across, and we're not just having it diffuse across into the lumen. How do we carry hydrogen ions in the blood? There was a buffering capacity that I remember. There was a particular ion that I talked about that buffered hydrogen ions. Bicarbonate ions. Okay? So bicarb is present in the blood, and it picks up hydrogen ions, and it carries it. And in that process of carrying it, you're going to see how we eliminate specifically in the proximal convoluted tubule. So I know this looks a little scary. It's because it is. And we'll see it over on the right. And I want to go specifically into this particular antiporter. Now, I won't test you guys on the extreme mechanism of how this takes place, but at least you can still gather some principles from here. And what I'm first going to start off with is, again, the antiporter. Here I'm going to have sodium going into the cell, and that's what I'm first going to start off with. And I'm going to end with hydrogen ions leaving. So the general principle here of what we want to do is we're going to be able to see hydrogen ions that are carried in the blood. And this is where we have bicarbonate. Bicarbonate, we need to replenish and get bicarbonate back into the blood. CO2 is something that's typically generated. If we have a bunch of hydrogen ions being produced, we know we're typically undergoing glycolysis, and we're probably going through a lot of glycolysis, and that's pushing a lot of lactic acid into the blood. That acid gets buffered by bicarbonate, and as a byproduct, we have a lot of CO2. And that CO2 gets diffused into the tubule cell. Not only is it coming from the peritubular capillary, it's also coming from the interstitial fluid. The third place of where it's being produced, we've got a lot of the cell producing a lot of CO2. Why is that? You guys already pointed out that the sodium-potassium pump is using ATP to fuel active transport. So if this cell has to continually generate ATP in order to transport sodium across, it's undergoing cellular respiration. And in the process of undergoing cellular respiration, we're producing a lot of CO2. So we've got CO2 coming from all over the place. And with CO2, once you get CO2 and you get water together, water that's already present in the cell, those two come together. So CO2 and water form carbonic acid. And carbonic acid dissociates into hydrogen ions and bicarbonate. So what's happening is that CO2 and H2O bind together. They form this weak acid called carbonic acid, and carbonic acid will split. And that split, where bicarb now diffuses down its concentration gradient back into the capillary. So that should make sense. If I've got a lot of CO2 in the blood, I've got a lot of hydrogen ions, what better way to pump bicarb back into the capillaries? Now what happens with that excess hydrogen ion that we split from that carbonic acid? That hydrogen ion then gets pumped out through the antiporter and gets secreted back into the tubule. So that should satisfy what we've just talked about. That's sodium reabsorption, which we saw, and hydrogen ion secretion. But that hydrogen ion isn't done yet. What we can do with that hydrogen ion is it also gets buffered. It gets buffered, and the one thing I neglected to mention here, that what catalyzes the reaction of CO2 to H2O, CO2 and H2O into carbonic acid, carbonic anhydrase. That shouldn't be new to you guys. That you've seen several times now. Now, there's also carbonic anhydrase that's present on the outside of the tubule cell. And when that hydrogen ion comes in, any of the bicarbonate that filtered out, all the bicarbonate that actually did get filtered out, let me reverse that. Any of the bicarbonate that does also get filtered out that's present in the lumen also gets buffered by bicarbonate. But that bicarbonate then forms carbonic acid. That splits off. You get now water and that CO2. That CO2 comes in and fuels this whole system again. So this is our way of, once again, driving more bicarbonate back into the blood. So in these slides, this really points out the apical side. This is what's happening on the apical side. This is what's happening on the basilar side. And this is what's happening in the cell. Rena, you had a question?
So how did you get the CO3 from, okay, so
Which
the
one?
nitrogen, no, on the bottom one.
Yeah.
That one, yeah, that one.
This CO2 right here?
The top one.
The
The H2, or the bicarb, yeah.
Where did that come from? That's what's already present in the lumen.
Oh, okay.
Yeah, so you have bicarbonate that's already present there. All we've talked about is the movement of sodium and the secretion of hydrogen ions. Going back to this earlier slide, let me go back to what's already present in the filtrate. Bicarb is right there. Okay. That answer your question?
Yeah.
So this process helps conserve that bicarbonate. Any of that bicarbonate that we would have lost in the filtrate also gets reabsorbed and pumped back into the capillary. It just has to go through and get transformed a couple times back into CO2 and water, and then back out through carbonic anhydrase. So in essence, carbonic anhydrase facilitates the reabsorption of bicarbonate across. Yes? Carbonic anhydrase facilitates the movement or the transport of bicarbonate from the lumen into the cell, back into the capillary. So when you look at these particular slides, what will help you look at this is start off with the main idea. Well, really not the main idea, but just look at the caption. HCO3 reabsorption. Locate where that bicarbonate is and follow the path. CO2 gets back into the cell. This is the intracellular mechanism where carbonic anhydrase converts it back into carbonic acid, splits it off into hydrogen ion, which flows right back out. Bicarbonate gets pushed back in and reabsorbed back into the capillary. I know it looks a little scary, but again, start off with what you're looking at. And here, the same process. Start with your sodium, end it with hydrogen ions. And once you kind of follow it once, twice, 3 times, 4 times, however number of times you get it, you'll see it. But the main idea, again, is to just look at how we shuttle things across the apical side. And as a change of pace, Look at the apical side. What do you guys notice about the apical side? Is it straight? No. It's got villi on it. And that the microvilli that are present on the apical side, increased surface area for absorption. The last thing we're going to see is passive transport. And the main idea behind passive transport is looking at electrochemical gradients. When we push sodium across, sodium has a positive charge. So if I'm pushing a positive charge into the cell and then pushing it out of the cell, I'm going to have other things follow as well. And what things am I going to be talking about? Chloride. Anytime I have a positive ion move along, you're going to have a chloride follow along with it. And sodium, NaCl, both combined together, table salt, right? This is our way of conserving salts. So the second most abundant ion would be chloride, the first being sodium. So the main idea here is utilizing an electrochemical gradient that allows the movement of other ions as well. So I don't go into the mechanisms behind this. I've talked about sodium. There's different mechanisms for each of these: potassium, calcium, magnesium, urea. I'm not going to get into those. But the general principle, sodium moves across, chloride will follow as well, and it'll move in between through the paracellular route or the transcellular route. Again, reabsorption that takes place down the line. So what's taking place in the first half? Movement of sodium. What's taking place in the last half are the other ions. So not only are we moving the ions, but we're also establishing an osmotic gradient. Again, general principle, move a bunch of salts, move a bunch of ions, we're going to get water that moves across as well. The one thing I won't get into too much detail here is urea. Urea kind of comes in as well. Urea, again, is a byproduct of the breakdown of amino acids. Again, not to go too much into detail again, but the structure of an amino acid has a carbon here, and this carbon is attached to a carboxylic group, and a functional group attached. And here the deamination involves the breaking apart of this particular bond, which releases NH2. And NH2 is our amine group, and then mix that with a hydrogen ion, and that creates NH3+. And that is ammonia. And ammonia then gets broken down into ammonium, and that ammonium, through this process, creates urea. So urea is a particularly small molecule. Basically what it's doing is it's trying to maintain this osmotic gradient. So I won't go into too much detail about it, but if you see reabsorption and you see secretion later on, it's a concept, if you ever want to get into more detail about it, it's called urea recycling. It's a way of being able to push water along and kind of hurry it up along. What we're talking about, here you can see a recap breaking all of this down. I won't go into too much detail again because this is basically a reiteration of everything we talked about. But the movement of sodium also allows the movement of other ions and other molecules. We use our sodium-potassium pump to pump things across. We get potassium that comes in as well through that sodium-potassium pump, but can also leave as well. And gets either pumped back in and then pumped right back out through leaky channels. There's a coupling that takes place between these two. So both salts, both sodium, calcium, glucose, amino acids, anytime we move ions, we move water as well. So it's safe to say that that osmosis is coupled with active and passive transport of all of these molecules. Key word that I want to illustrate here is coupled. They're kind of married together through that general principle. Other things that you'll see that's also taking place in the proximal convoluted tubule, any lipid-soluble substances moves through this transcellular route. Anything also moves through the paracellular route as well. Again, 65% absorbed in the proximal convoluted tubule. Most of your glucose, most of your vitamins, your amino acids, proximal convoluted tubule. What we're going to talk about next in the loop of Henle is the reabsorption of salt and water. But what's characteristic about the loop of Henle is an uncoupling of water movement with sodium and chloride. And that's the main idea of what's happening in the loop of Henle. You saw coupling through here, so that main mechanism that I talked about, water moving along with transport of stuff. Here in the loop of Henle, the characteristic, and I won't go into the makeup of the cells, but namely the descending limb, which has cuboidal epithelial cells, is impermeable to ions. It doesn't have any of the channels, it doesn't have any of the co-transporters that we saw prior. So the only thing that can move down the descending limb is water. Okay? And that water is due to the aquaporins that are present in the descending limb. So how much are we absorbing or reabsorbing? How much water are we reabsorbing? About 15%. 65% in the proximal convoluted tubule. 15% here. By the time we make it down to the loop, we've now reabsorbed 80% of our water. We still have this leftover that we still need to account for, which we'll talk about on the next slides over. The ascending limb, on the other hand, is impermeable to water. And if you look at the structure, and you'll see in any book that you ever look at, they'll draw the thicker squamous epithelial cells that are present as the tubule cells. And this thick layer of epithelial cells makes it impermeable to water, but it allows the movement of sodium and chloride. So again, that uncoupling, water moves down, sodium and chloride move out. And what do we notice? Here you might see the numbers that are also present. You'll see the numbers, and these numbers represent, technically here, this is the osmolality, but osmolarity, same thing pretty much when you're looking at water. Osmolarity, again, references the concentration of the solute with that of the volume of water. So something that has a lot of solute and very low volume is going to be hyperosmotic. And that hyperosmotic is going to be a fairly high osmolarity. So what you're going to be able to see is when we go from 300 to 500, 700, and 900 to 1100, we see a rise in osmolarity. What did we lose as we move down the descending tubule?
Water.
We lose water, right? So what's happening to the osmolarity? There's no change in the solute concentration. Why? Because it's completely impermeable to it. What we're losing is water volume. So a decrease in the volume is going to cause an increase in the osmolarity. In other words, if you can think of osmolarity, and let's substitute a better word for it, how salty is that urine getting? So if you can sit there and kind of like taste the urine right there and then like kind of taste it over here, it gets saltier as we move down. And it gets saltier because we're removing water. Take any salty concentration, take salt water and just remove the water out of it, the water's going to get saltier and saltier. And that's exactly what's happening as we move down the length of the descending tubule. If we look at the ascending tubule, what's happening in the ascending tubule? Water remains on the inside and it doesn't move. It's completely impermeable to water transport. Now what I'm doing is now I have this salty filtrate. I'm now able to pull sodium and I'm pulling chloride. So now I go from a hyperosmotic filtrate right here. As I start removing salts from it, that's going to bring the osmolarity down. Why is that? Because now I'm reducing the concentration of solutes in the filtrate. So think about, in effect, what am I really doing? Again, uncoupled. I'm removing water, and now I'm removing salt. So by the time I make my way up to the top of the ascending tubule, I almost have the same concentration, but I've removed a good amount of water and I've removed a good amount of salt. Does that make sense?
So you had the same concentration at the beginning?
Pretty much. But same concentration, but concentration just tells you the composition of the filtrate, but it doesn't, what it means that by the time I get to this point, I've got also a lot less filtrate. Think about that for a second. I removed water, I removed salt. I'm gonna have a lot less of it. So, in this process, I've already mentioned that water moves out through aquaporins. I have to look at how we move sodium and chloride. Because sodium and chloride are going to be the big things that we're trying to move across. So we have to look at the symporters that are present in the ascending limb. Again, we're focusing on the right side, the ascending limb. And how do we get salts across? The slides here we'll go into. We're going to, again, looking more images here, and just to quickly reorient you. You're going to be able to see the vasa recta. Again, here you saw paratubular capillaries in the PCT. It's now called the vasa recta in the loop of Henle. But nonetheless, reabsorption still remains the same. It's still moving in that direction from the tubule over to vasa recta. You're still going to have the same processes, paracellular, transcellular. Now, focusing on the transcellular side, we have to look at what's present. In order to get those ions across, we have to see what's on the apical side as well as the basilar side. Nothing new with the basilar side. You already pointed out earlier, sodium-potassium pump on the basilar side utilizes ATP to push sodium out. The symporter that we have present on the apical side, we have salt, namely sodium, 2 chloride ions, and a potassium ion. 2 positive ions, 2 anions. Moving across, so still electrically neutral as we move across. Salt we want to move across, same, nothing changes there. Sodium moves down the same channel. Chloride also moves across, and it moves down its concentration gradient once again. Potassium, on the other hand, potassium gets reabsorbed, but it also gets channeled right back out. And this is where we're going to start to get into whether we're going to reabsorb potassium or get rid of it. Now, when we get into the distal convoluted tubule, you're going to see that potassium is kind of one of the exceptions. I did ask a quiz question way back when about potassium. Out of all the things that we looked at, we want to be able to conserve sodium, we want to conserve chloride. Those are the ions that are present on the extracellular fluid. But there's one thing that I mentioned we didn't want high in the extracellular fluid, and that was potassium. Remember when I mentioned before, like people with cardiac issues, they need to monitor their potassium intake because they can't really get rid of potassium, or they produce extra potassium, which we call hyperkalemia. Here we need to regulate our potassium. Potassium we want inside the cell, we don't want it on the outside of the cell. And what helps maintain that? Sodium potassium pumps shuttles it in, but we don't want it too much on the outside. So I don't want to get too much into that, but that's really the main idea. Leakage channels for chloride, because chloride we want to definitely conserve. Sodium we definitely want to conserve. And then the electrochemical drive, same thing. The movement of potassium back out, right? So I get 1 positive ion in, 2 anions in, so, potassium back out. We're going to get an electrical chemical drive that allow other ions to move through the paracellular route in order to maintain electrical neutral movement of ions across.
You said you wanted potassium outside the cell, right?
You want a potassium
Outside?
out? No, potassium
Inside.
you don't want. You want it inside the cell.
Okay.
So while the sodium potassium pumps help shuttle potassium in, we do need to get rid of excess potassium. So in these cases where you see sodium going across, you see chloride going across, potassium might go back into the lumen. It's our way of maintaining that balance. Out of all the ions, yes, potassium, we want a lower amount of it. Let's now look at the whole concept of countercurrent mechanisms. The whole idea behind countercurrent is one goes this way, the other goes that way. That's the whole idea behind countercurrent mechanisms. And we're going to see different ways in which we're going to see one way and another way. Countercurrent, one current going this way and the counter current going the opposite way. We'll look at countercurrent flow, countercurrent exchange, and countercurrent multiplication. They all start with countercurrent, but yet they all, for the most part, talk about a different principle. The first one, countercurrent flow, we already talked about. In this slide, you saw, as I described countercurrent flow. Countercurrent flow is basically showing that we're going from 300 osmolarity down to 100, so about 1,000 to 1,200 osmolarity by the time we make our way down. And that mechanism we already illustrated. Why? Because we're removing water. This is where I talk about removing water, and now you get this salty filtrate. So we go from about an isotonic, hypotonic, down to hyperosmotic or hypertonic. And then when we go from hypertonic, we go back and we're moving all the way back up. This is basically illustrating that this countercurrent is we're going down from low down to high and high back down to low. So again, that's due to the uncoupling of the movement of water down the descending limb and the movement of sodium and chloride up the ascending limb. You had a half question.
Yeah, I did. You went from isotonic to hypertonic and then from hypertonic to isotonic.
Yep, that's basically it. So what does that mean for the medulla? Medulla splits this loop somewhere along down the line. That means if you were to look at the cortical regions of the kidney, it's not as salty as if you were to get down to the medullary area of the kidney. So if you were to taste a piece of the kidney, the saltiest part would be on the inside. And the normal tasting part, actually I don't know, I don't eat kidneys, but nonetheless, the whole idea behind it is it's definitely saltier, and it's saltier due to the hypertonic or hyperosmolarity of the medullary regions. That's countercurrent flow. Countercurrent exchange is now taking that same limb, descending, ascending limb, and looking at the other portion that we need to account for is the directional of the blood flow. And you can see that on the middle of the slide over, countercurrent exchange. Filtrate moves down the descending limb and back up the ascending limb. Countercurrent exchange shows that the vasa recta actually flow the opposite way. So the capillaries that wrap around the ascending tubule are the descending portions of the vasa recta. So, in essence, filtrate movement, blood movement. What's the advantage of that? So in this case, where I have...
That's why you drew the capillaries that way?
Yes, exactly. That's why I drew it kind of off. How far back? Somewhere far back, right over here. I kind of drew it the opposite way. That's countercurrent exchange. So thank you for pointing that out. That is exactly what I wanted to get at, is that here we have hypotonic filtrate. I'm also going to have the same token, also kind of a non-salty hypoosmotic blood. And that's still going to allow the salts that move, that we're able to transport across the ascending limb, to get absorbed by the blood. So as the blood comes down, it's going to get saltier and saltier as it's picking up the salts from the ascending limb. So now if I were to come up here and kind of taste that blood would be salty. And that saltiness of the blood, again, think of that U tube that I drew earlier. Now I got a bunch of solutes on one side. I've got all my solutes of salt in that blood. That's going to drive the water that's moving out, that's being transported out of the descending limb to move it into that salty blood. So this whole mechanism allows that blood to quickly pick it up from the interstitium. We've got a salty medullary interstitium, and that comes in and picks up the excess salts. All right? And it picks up the salt down in the descending and picks up water in the ascending portions of the vasa recta. That's countercurrent exchange. The other portion is countercurrent multiplication. And the whole idea behind countercurrent multiplication is just looking at the saltiness of the medullary. And due to that saltiness, and this is where urea kind of comes a little bit into play, the saltiness in here just creates that osmotic gradient. Water comes in, and even though we're losing water in the descending region, it's not changing the osmolarity so much in the interstitial fluid because it's getting picked up by the saltiness of the blood in the vasa recta. So typically it would be salty here, and that allows for this osmotic gradient. It's all part of this sort of transition as we get water back in, reabsorbed back into the vasa recta. So what's going to happen is that when we set up that saltiness right here, this is going to play a role in everything from the distal convoluted tubule down to the collecting duct. As we start getting fluid that moves its way down, as long as I have channels for water, water is going to want to move into a salty area. But the big thing that I did point out is that we have to have channels that are opened. And what do you think are going to open those channels? You might not be able to answer it yet, but you'll definitely be able to answer it later. The key idea are your hormones, of which we'll talk about, ADH and aldosterone. This is countercurrent multiplication, same thing that I talked about earlier. This leads itself into the whole idea behind the formation of urine. So as I mentioned earlier, by the time we get up to the distal convoluted tubule, which we'll pick up next, we'll continue along with reabsorption. Now, reabsorption in the distal convoluted tubule, we're still going to look at the same things. Sodium, we'll look at potassium, we'll look at chloride. And I know we're going to be looking at more and more of these symporters and antiporters. I won't get into too much detail about it, but the main idea behind these, this is still our way of collecting sodium chloride. We're going to start to see the effective hormones that will allow additional transport of other ions, namely our parathyroid hormone, and we'll see ADH come in as well as aldosterone. The two types of specialized cells that we have in the collecting duct, again, collecting duct would be after the distal convoluted tubule, is our principal cells, and principal cells reabsorb sodium and secrete potassium. Interstitial cells reabsorb potassium as well as bicarbonate and secrete potassium. Principal cells, again, following the principle of reabsorption from the lumen over to the peritubular capillary. Again, peritubular capillary, because we're moving back into the distal convoluted tubule to the collecting duct. Principal cells, in general, if I can quickly sum up the principal cells basically will be utilizing leaky channels on the apical side and involve leaky potassium channels on the other. So if you were to take a look at potassium, remember what I mentioned about our principal cells. Two things we're going to be looking at are sodium and potassium. Sodium, that same mechanism doesn't change. Sodium in through leaky, sodium gets pumped out, moves down its concentration gradient. There's no change in that. That's not new news to you. What is new are the presence of leaky potassium channels on both sides. Why is that? So potassium that's pumped in can either move back out into the interstitial fluid or move back into the lumen to be secreted. So the principal cells are the main source of potassium regulation. It all depends on whether you need to hold on to excess potassium, in which case you're going to be reabsorbing it. So in the case here on the basal lateral side, you'll see these leaky potassium channels that allow potassium to leak right back out. These are the same leaky potassium channels that you would see in the heart cells. It's the same makeup. Or you'd also see potassium on the apical side to get secreted back out. So this is how we maintain. And again, if you ever get a urinalysis done, there's a narrow window of where you want your potassium to be. If you've got too much of it, secrete it back out. if you don't have enough potassium, you're going to reabsorb it back in. Now, the other thing about principal cells is that they're also stimulated by not only our ADH cells, our antidiuretic hormone, which we'll take a look at later, but they're also stimulated by aldosterone. Now, ADH governs the movement of water. Again, antidiuretic hormone governs the movement of water. Aldosterone regulates the movement of sodium and potassium. Remember what we mentioned about renin, angiotensin, aldosterone. We looked at three main things that were high on the outside. We had sodium, we had chloride, and we had high intracellular potassium. When we regulate with aldosterone in that system, the RAS system, we want to be able to conserve sodium, we want to conserve chloride, but also we want to decrease the amount of potassium. And also, through that virtue, because we're reabsorbing sodium and chloride, we're also going to see the movement of water as well. So with that principle, you'll see our principal cells that will govern that due to aldosterone. These cells have receptors for aldosterone that will, again, receptors that are present on the basilar side, that will respond to our hormones to help adjust either potassium and then water, which you'll see later. Our intercalated cells, again, looking at intercalated cells, reabsorption of potassium and bicarbonate and secretion of hydrogen ions. So these, if principal cells were more governed towards potassium regulation, intercalated cells govern acid-base regulation. They secrete hydrogen and they also reabsorb bicarbonate. Bicarbonate, again, important for buffering. So take a look at what's happening with our intercalated cells. Again, we're wanting to secrete hydrogen ions and reabsorb bicarbonate. This whole system right here, again, I'm not going to go into that same amount of detail that I did earlier, but it's the same mechanism. In this case, you get CO2 and water. They come together to form carbonic acid. Hydrogen ion gets secreted out. Bicarb gets sent back into the peritubular capillary. So in this process, what we're able to do is to consistently pump hydrogen ions out. We're utilizing this pump to secrete hydrogen ions. It's using ATP, and this way we can now pump hydrogen ions against its concentration gradient. Remember, we can have a lot of acid present, and throughout that process, as you learn, we're pumping out hydrogen ions that are in excess into the lumen. So we're starting to get some acidity in there, and that acidity gets continually pumped out as we pump out hydrogen ions against its gradient in order to conserve bicarbonate. So what happens with that hydrogen ion? That hydrogen ion can then merge with ammonia to create ammonium. It can also get buffered by phosphate, which phosphate will then turn into phosphoric acid. Very, very minute phosphoric acid, but nonetheless, all stuff that we're going to secrete out in our urine. So the main idea behind the intercalated discs, secrete hydrogen ions to concentrate that acids into the urine. Diluted urine. So how do we produce dilute urine? So think about a case in which you would create a bunch of dilute urine. How do you get really watery pee? Drink a lot of water, right? Drink a lot of water, and in that process, what we're doing is we're not able to reabsorb as much water. So in this case, remember what is involved in the case of excess water. We've talked about reabsorption that takes place in the proximal convoluted tubule. Now 80% of water is reabsorbed by the time we get down to the bottom of the loop of Henle. We're going to come back up where we have the distal convoluted tubule. Right at this point is where we start the distal convoluted tubule, and you'll notice the osmolarity about 100, and then the osmolarity going back down to 90, down to 80, down to 70, down to 65. Now, why is the osmolarity not changing as much? In fact, it's going down a little bit. let me rephrase that. Why are we not reabsorbing water in this blue area? We know this brown area right here was impermeable to water. And not just due to the ascending limb of the loop of Henle. But by the time we get down to the blue area, you notice that we're not reabsorbing water. That should make sense because you just drank a bunch of water. Why would I need to reabsorb it? The distal convoluted tubule leads back into the glomerulus and will say, well, no, it looks like I've got enough salt in there. And in fact, by the time I get down to the collecting tubule, I probably won't want to reabsorb as much water. So in other words, the area in the blue is impermeable to water because we don't activate any of the water channels in the collecting duct. What would activate those water channels? ADH, antidiuretic hormone. And in what cases would you see antidiuretic hormone being secreted? When you're dehydrated. When you don't take enough water in, you have blood loss. Right? I want to activate the water channels, our aquaporins. I want to activate them to reabsorb more water. Hence, an antidiuretic hormone. Hence, if you were to be dehydrated, you would want to reabsorb as much water as possible. So let's take a look at what's happening in the concentrated urine. Concentrated urine, we're going to go back up to about 100 to 200 osmolarity back at the distal convoluted tubule. And by the time we make it down to the collecting duct, ADH, released by the posterior pituitary, which we already know, responding to low blood flow, results in water reabsorption in the principal cells of the distal convoluted tubule and the collecting duct. Exactly what I was saying earlier is that activating the aquaporins on the principal cells. I'm going up to these principal cells right here and saying, once ADH comes in and binds to this area, it's going to actually turn on them on, to now open up these aquaporins that allow water to move in. So this facilitates, that ADH binding to facilitates the movement of water. Otherwise, water would just normally diffuse across as we get salt to move in. What better way to move water faster than to create these channels that will shuttle water quickly through? And that's exactly what ADH does. It acts directly on those principal cells to open up those channels. And therefore, we're going from a 300 osmolarity, and we're going to start pulling water out. And pulling water out is going to make the urine saltier. In other words, it's going to concentrate that urine even more. So that by the time we get to the end of the collecting duct, bless you, we have very hyperosmolar urine. Very high in the amount of stuff that we secreted out, but very low in the volume of water.
So ADH only binds to receptors when we are like, when we're dehydrated?
Yeah, so the key thing is that we have osmoreceptors that feed back into the hypothalamus. And the hypothalamus then triggers the posterior pituitary to release ADH. That ADH then flows through the circulatory system where it activates also in the kidneys. It activates in the kidneys right on the principal cells to allow water to get reabsorbed. Does that help?
Yes.
Okay. So in this case, what's happening? Urine becomes more and more concentrated and water loss becomes minimalized. Again, the loss or the low blood volume or dehydration results in ADH that activates water reabsorption in the collecting duct. As opposed to in dilute urine, If I've got a lot of water, the osmoreceptors aren't firing as much. Therefore, no ADH. No ADH turns off all the aquaporins. Therefore, dilute urine getting released. The principle behind this, and there's a lot of text here, tubular reabsorption is basically saying if you've got too much stuff and the nephron can't absorb it, well, it's gonna get peed out. That's basically all it's saying. If higher than normal amounts of a substance are present in the filtrate, then the renal threshold for reabsorption of that substance may be surpassed. That's all that's saying, is that if we can't reabsorb it fast enough, we eliminate it through urination. That's what's going to make its way down the PCT, down the loop, down the distal convoluted tubule, back down over here. If we cannot reabsorb it fast enough because we have so much of it, we end up peeing it out. And now let me go back to diabetes mellitus. Now what we notice about diabetes, type 2 diabetes, is, and did I tell you guys about the origin of the term diabetes mellitus? Sweet urine. Old ways of, obviously way back in the day, they haven't been able to formally do a glucose stick. Like before they were able to diagnose high blood sugar, they couldn't really, they didn't have glucose sticks back then. The only way of being able to tell is that they were able to tell by the sweetness of the smell of the urine. So one of the ways they had to be able to tell what was in your urine was to taste a little sweet. Technically, diabetes mellitus means sweet urine. So why is that? Why did it taste sweet? All starts at this point. High blood sugar, you've got a lot of glucose in the sugar, right? And we already talked about the mechanism of diabetes mellitus, is that our insulin receptors aren't able to receive insulin as well. And therefore, insulin can't pull glucose into the muscle, it can't pull glucose into the liver, and therefore it's just sitting in the blood. And it's circulating through, and it's changing the osmolarity of the blood. And the osmoreceptors are picking that up and saying, wow, you've got some really thick blood. It's going to drive up thirst, it's going to drive up water retention. Remember, saltier, or not maybe saltier, but the more solutes in the blood, water is going to now flow in as well. Hence why people with diabetes, if uncontrolled, can edema. A lot of edema, a lot of water retention. Not only water retention, and always feeling thirsty. And not only that, they also need to pee a lot. They constantly have the feeling the need to pee. So what's happening is that a bunch of glucose is getting filtered out. And it's making its way through. A good amount of the glucose is absorbed in the PCT. But because there's so much glucose, you can't reabsorb it fast enough that by the time it makes its way through, it's secreted out as urine. Hence the tasty. Well, that was some tasty.
Wow.
Sweet urine, okay? So that's basically where the whole mechanism that takes place, and that's exactly what's being described by this slide, is that the renal threshold, at least for the glucose transporters in the proximal convoluted tubule, is maxed out. We can't absorb it fast enough. And looking at the renal threshold, we can absorb about 180 to 200 milligrams per deciliter, 100 to 200 milligrams of deciliter of blood. So this is the whole concept of spillover, glucose spillover into the urine. Okay? And that's why we regulate glucose on the blood level. Pancreas should be secreting enough insulin. Insulin should be coming in to help reabsorb the glucose into the liver as well as the muscle, not into the kidneys. That's a lot of wasted glucose. And as we were able to talk about earlier, you saw the different aspects as to the other symptoms of diabetes. Now, acid-base regulation all boils down to how we can maintain our pH. And the pH of blood, again, is between
7.35.
7.35 to 7.45. 100% of you guys should be able to get that right, because I know you guys got that right on the test. We have other mechanisms that we're able to maintain acid-base regulation. And you can see by now, we have the lungs, we have the kidneys, and the digestive system. We didn't talk so much about the skin, but at least these three mechanisms help regulate that balance. Lungs eliminate CO2. And remember what we mentioned about CO2 and water produce, through carbonic anhydrase, produce weak carbonic acid, which also then disassociates to hydrogen ion and HCO3. Bicarbonate, you saw, we were able to reabsorb. Hydrogen ions, we were able to secrete. Carbonic acid, and then CO2 and H2O. The lungs eliminate CO2. So if I eliminate CO2 from this equation, that's going to drive this equation through the Le Chatelier's principle, which is going to move from hydrogen ions to carbonic acid to moving over this way. So this elimination of CO2 is a very fast response to maintain our bicarbonate stores. Blow off excess CO2, and you can restore your bicarbonate stores. That'll help buffer any of the hydrogen ions that are accumulating. Nonetheless, this is our way, a very fast response. Whenever we start thinking about exercise, for instance, whenever we start exercising, and in the case of, let's say, cardiovascular exercise, you're undergoing cellular respiration, you're producing hydrogen ions, that's getting buffered, and that's going to come in and get buffered, and we blow off that excess CO2. That should make sense. These are all byproducts of cellular respiration. Now, what's happening to the others of hydrogen ions from this equation? If we're conserving bicarbonate, what are we doing with this, with our hydrogen ions? We're eliminating that through the kidneys. And that takes a little bit longer. These aren't very fast responses, because this is all through the part of secretion. Now, the digestive system and the skin also help as well, but not to the biggest extent, but the fastest response is our lungs. So acid-base regulation, a lot of that takes place in our lungs. And again, all through excess CO2. Our hormones that are involved, we're going to be able to look at our hormones that are going to help in the process of ion regulation. So looking at sodium, chloride, calcium, water, as well as potassium, we'll be able to look at how these all play a role. Antidiuretic hormones, the first one we'll talk about. ADH, we already mentioned, targets the principal cells in the DCT and the collecting duct. That we saw with concentrated urine versus dilute urine. In order to concentrate our urine, we have to utilize ADH that will allow for water reabsorption. Renin, angiotensin, aldosterone. While ADH has a direct effect on the aquaporins, or in the water channels, angiotensin and aldosterone have an indirect effect on water. You not only have seen this in our endocrine system, you've also seen it in our cardiovascular section. Now, revisiting it again is looking at our juxtaglomerular cells. Juxtaglomerular cells are the cells, again, that are leading into the glomerulus. This is the afferent arteriole. These cells will detect the blood flow into the glomerulus. And if there's decreased blood flow, we saw renin. Renin was secreted. And that renin comes in and finds angiotensinogen that's floating around the blood that's been released by the liver. Again, one of the non-digestive proteins that I quizzed you guys on. Angiotensinogen comes in, it's cleaved by renin, and now you have angiotensin 1 that's converted to angiotensin 2 by way of our ACE enzyme in the lungs. ACE, being our angiotensin converting enzyme, converts it into angiotensin 2. Angiotensin 2 then comes back and vasoconstricts our arterioles. Okay. Where does it do that? Angiotensin 2 comes in and vasoconstricts our glomerulus. So just kind of quickly drawing out our glomerulus. If this is our efferent arteriole. Due to the smooth muscles, afferent arteriole. This is afferent, this is efferent. The smooth muscles that wrap around are stretch receptors. And they come in, and if they don't detect enough blood pressure or blood flow into the glomerulus, that decreased stretching comes in and results in the production of angiotensin II, which then causes vasoconstriction. And that vasoconstriction then increases the pressure to allow for normal filtration to take place. Okay. Where else does angiotensin II come in? Increases blood volume by increasing reabsorption of water and electrolytes in the PCT. So that's where it comes in and plays a role over here to increase water reabsorption. And also stimulates the adrenal cortex to release aldosterone. Angiotensin comes into the adrenal cortex, and that's where we get aldosterone. And where does aldosterone come in? This is aldosterone that comes in and affects the principal cells. ADH affects the same principal cells, but results in the production of water channels. Aldosterone stimulates the principal cells to reabsorb more sodium and chloride and secrete more potassium, just like I described earlier on this slide over. We want to be able to reabsorb sodium and potassium and secrete potassium, sodium and chloride, and secrete potassium. And again, general principle is that if you're moving salts, sodium and chloride, water is going to follow as well. So water will then move and get reabsorbed as well because you're moving salts into, you're reabsorbing, water will move through. So what would be the most effective way of getting water into the peritubular capillaries? Is to activate with ADH, low blood volume, and low salt content. Right? So the low salt content comes in, and then that plays a role in water reabsorption through the principal cells. So you have ADH and you have aldosterone that allows water movement through, and that's going to help regulate and maintain normal levels of our glomerular filtration rate, and then optimal water reabsorption throughout the nephron. So all of that boils down to this slide over here, which helps summarize everything that we've mentioned so far. Again, looking at this, compare this to the first slide that I talked about that illustrated reabsorption in green and secretion, I believe, in blue. And you can see each of the breakdown of where water, our salt, our potassium, our chloride, and bicarbonate, all the big things that we talked about so far, the reabsorption of these ions throughout in order to conserve. Where you saw the biggest variation was in the distal convoluted tubule and the collecting duct. All that was related to our water reabsorption as well as our sodium and chloride due to ADH and aldosterone. And that ultimately affects the concentration of our urine. You also saw the secretion of our hydrogen, you saw reabsorption of bicarbonate as part of our acid-base regulation of that blood to tie in. And then any excess that we aren't able to reabsorb gets secreted as well. And that was our renal threshold. So all that boils down to the whole purpose of the nephron and how that plays a major role and why the kidneys are so important in maintaining homeostatic values. Whatever we're not able to absorb, get rid of it. Okay? And if we need to reabsorb more water and salts, we have the ability to do so to maintain normal volumes of water, and then most importantly, maintain the amount of blood volume and blood pressure for optimal functioning. Last two slides I want to talk about, urine. Water is mostly your urine right there. Anything else that's supposed to be found? Again, things in a urinalysis is basically looking up the content of your urine to make sure that you have the right amount of ions. Anything in excess will affect the what's released in your urine. So not only is it very useful to find out what's in your blood through blood labs, it's also really useful to find out what's in a urinalysis, because these are ways of getting rid of, good way of cross-comparing what's happening in the blood as well as your urine. Anything else present, bacteria, albumin, protein, glucose, white blood cells, and red blood cells are things that shouldn't be excreted. Why? Because we saw albumin, protein, White blood cells and red blood cells, they should not be present due to the glomerulus. Those filtration slits prevent those things from being secreted. If you find it in the urine, something's up. Okay. If white blood cells, obviously you've got excess of white blood cells. If you've got excess of glucose, again, something to do with your blood sugar, and something might be wrong with your insulin or your pancreas. So these are good diagnostic clinical tools to find out what systems might be malfunctioning. The other things too is to measure renal function is to take a look at your nitrogen balance. And all that boils down to is how much you're breaking down proteins from catabolic reactions. Blood urea nitrogen is something that's present in the urinalysis that tells you how much nitrogenous waste you have. Anytime you break down your amino acids, you get urea. And urea is going to contain nitrogen in it, those amine groups. And that blood urea nitrogen lets us know how much of the catabolic reactions are you going through. How much are you breaking down your protein? Not only are you breaking down your protein, where's that protein source? Is it from your muscle? We can couple that with levels of creatinine. Again, creatinine being present in skeletal muscle as an ATP storage. If you find creatinine levels in the blood, you may want to ask that person, are you taking creatine? Do you have anything else going on? I see a lot high levels of creatinine. This must mean you were breaking down protein. You're breaking down muscle, skeletal muscle. These are things in the cases of ever looking at rhabdomyolysis. It's basically the presence of blood in reddish urine. It's a heavy breakdown of skeletal muscle. It's part of the inflammatory process that's on the extreme side. So these are ways that they can come in and measure these tests. So out of all the things that we looked at, sodium, chloride, these, they can look at nitrogenous balance and determine the content of that to give you an overall look at the human body. With that, that covers pretty much everything I ever need to talk to you about. I will see you guys next Tuesday.